Hello, welcome. How's everyone doing? Lots of questions in the chat that we need to answer already. Just before we start, welcome everybody. Let's just clear up who this course is for. I think that's really important. So this is for AQA. This is AQA, okay? Which you guys should have been told. So it is tailored to AQA. Now, lots of you are obviously doing different exam boards. And what I've been saying in the chat is a lot of the content is the same, but the order of topics is different. So you've got to be aware of your own specification and what is on your paper one, right? Because I don't want you panicking and revising stuff tonight that is actually going to be on your paper two if you do Edexcel, for example. So you're welcome to stay. A lot of the content is the same, but just be aware that some of the questions might not be relevant. Okay. Hello. How long is this course? So I've planned it to be 3 hours, but I already know that for you people that do separate science or triple science, it's going to run over because I've chosen some separate science only questions to do in the last half hour. So if you're doing separate science or triple science, as you might call it, it's going to be at least 7:30, I would say. This is perfect for foundation and higher because what a lot of you don't realize is the content is pretty much the same. I think there's like so much confusion, like all of the combined content is on separates as well, right? And all of the foundation content, the higher students have to know as well. So it's suitable for combined, separates, higher, foundation, AQA. Okay, this is going to be recorded. You can pause it, you can rewind it, you can watch it back later, you can watch it tomorrow morning. And we are going to be doing some practicals, microscope practical. I think I've got osmosis practical. I might do a little bit on just general how to plan a practical. So yeah, should we get started? Let's get started. Guys, we've got loads of questions to get through. We're covering questions from all parts of the paper one AQA specification, including cells, organization, immunity, and bioenergetics, okay? Let's get going. Now, in order to not be passive, which someone said in the chat, I recommend that you put your answers into the chat or you write your answers down in your workbook or just on a piece of paper if you've not printed it. And then obviously ask me questions in the chat as well. And I will try and answer as many as I can. Let's go. I can see answers already. This is fantastic. So A is the nucleus and B is the cell membrane. Because if you look really carefully, B, I know it's quite hard to see, isn't it? But B is pointing to the inside layer and the cell membrane is inside the cell wall, okay? The cell wall's on the outside of a plant cell, the cell membrane's on the inside, we should know that. Structure C is a chloroplast. This is a plant cell, it's a palisade cell. What is the function of a chloroplast? 2 marks though, guys. So always look tomorrow at the number of marks a question is worth. Super important. So you need to say 2 things here. Okay, Eva says absorbs light energy for photosynthesis. Eva, what do chloroplasts contain? Because we should get that down as well. Because what absorbs the light energy? Okay, good, Freddie. Chloroplasts contain chlorophyll, which absorbs light energy. And then we can say for mark point 2 for photosynthesis. Now, these are easy questions at the minute, right? This could be foundation, this could be on higher, but we should know they contain chlorophyll that absorbs light energy, and that's the site of photosynthesis. Okay, cool. I'm loving your answers so far. Let's keep it going. Let's keep the pace. Yes, triple and combined. All combined content is on triple. Like everything combined scientists learn, triple scientists learn as well. And we'll do some triple only questions at the end. Is this a predicted paper she's doing? No, but, and I should have said this is important, I have looked back at all the previous papers and I have chosen questions specifically on topics that I think are more likely to come up, if that makes sense. But it's not a predicted paper. I don't have a crystal ball, but I have chosen these questions for a reason. Let's complete the table. We need to give 2 more differences between a plant cell and an animal cell. Let's get our answers into the chat, please. We are doing A-level revision. We've got cram courses for A-level. We have. Right, we've already been given one, so obviously we can't repeat that. Don't be daft. Plant cells have chloroplasts, animal cells don't. Plant cells have a cell wall. Animal cells, no cell wall. And this is really nice, isn't it, when they give you a table, because it makes you say the differences. If they don't give you a table, make sure your answers are comparative. And what I mean by that is you're writing plant cells have a cell wall, whereas animal cells don't. But this is nice and easy because they've given us a table. And well done, everybody. Plant cells have a permanent vacuole. And animal cells, there's no permanent vacuole. Or you could say large vacuole. You could say plant cells have a large vacuole. Animal cells do not have a large vacuole. Perfect. Well done. This is AQA focused. A lot of the content is the same. You'll notice with Edexcel, like cells, right? It's the same. But remember, the order of topics is different. You can draw tables in exams, yes. If it's a question about differences, it can be quite handy to put your answer into a table so that you make those differences really clear. Yeah, I would say permanent vacuole. I would make it clear, because actually animal cells can have what we call temporary vacuoles. So just to make sure you get the mark, I would say a plant cell has a permanent vacuole and an animal cell does not. They both have ribosomes, Alex, so I wouldn't go for that one. Okay, describe 4 differences between a bacterial cell and a plant cell. So here you go. Here, this is a bigger question. It's 4 marks. It's harder. They haven't given us a table. So we could do our own table for sure. And I'm going to do it like that. And you could do this in your exam. Because if you do your own table, then you are bound to get marks, because you're going to have to make direct comparisons, right? Make the differences clear. You don't have to do a table, but if you don't do a table, I would use the word whereas in my answer. So let's get some answers into the chat. Bacterial cell is smaller in size, whereas a plant cell is larger in size. You see how the table helps me to make my differences really clear? Bacterial cells, someone said no chloroplasts, whereas the plant cell has chloroplasts. Or most plant cells have chloroplasts, so that would be accepted. Don't just say the plant cell has chloroplasts full stop. You would have to say whereas the bacterial cell doesn't. Do you understand what I'm saying there? Oh, the nucleus. Yeah, that's probably the most obvious one. I should have talked about the nucleus first, shouldn't I? So the plant cell has a nucleus and the bacterial cell, no nucleus, or you could say the DNA is just free in the cytoplasm. What about mitochondria? The bacterial cell can have plasmids, yeah. They don't all, but you can say it, because you don't actually learn that till A level. May have plasmids versus no plasmids. Or they did allow just the bacterial cell has plasmids, the plant cell doesn't have plasmids. Z and Ns, they both have ribosomes, so be careful. Ooh, mitochondria, yes, well done. The plant cell has mitochondria, whereas the bacterial cell, no mitochondria. Excellent work. I mean, we've defos got 4. We've defo got 4. Yeah, the rings of DNA are called the plasmids, so I've already got that one. Flagella may have flagella. Do all bacterial cells have flagella? Not all. So may have flagella, no flagella. May have a slime capsule. Sorry, my writing is going to get bad here, isn't it? No slime capsule. But be a little bit careful. Not all bacteria have flagella, right? Not all of them have a slime capsule. A slime capsule is like around the outside of some bacterial cells, outside the cell wall. Flagella are almost like tails, aren't they? They're used for movement. A sperm cell has a flagellum, right? Permanent vacuole versus no permanent vacuole. Yeah, plant cells have a permanent vacuole. Bacterial cells don't. I've put sizes at the top because you could say that. Mitochondria are the site of aerobic respiration. Multicell, you can't have that one. So someone's just said in the chat, bacterial cell is unicellular, plant cell is multicellular. But the question is about the cells. It's the differences between a cell and a cell. So you can't say that and get a mark. Plasmids are just small loops of DNA that bacterial cells have. Guys, can I ask you one question before we move on? You should know they both have cell walls, but what is a plant cell wall made of? What is a plant cell wall made of? We should know this. Perfect. Cellulose. Now, we need to be super careful here because bacterial cells also have a cell wall. So that is not a difference, but you could have got a mark for saying they have a cell wall, but it's not made of cellulose. You could have said that. I doubt you would have done, but yeah, it's a different substance, isn't it? It's actually called murin. Don't need to know that. But you could have said, look, they both have cell walls. The plant cell wall is made of cellulose. The bacterial cell wall is made of something else, peptidoglycan or murin. That's a difference. Not lignin, Tony. Lignin is found in the walls of the xylem. Okay, and it helps to keep the xylem vessel open so water can be transported. Don't forget. Yeah, Rosa, very good point, Rosa. Oh, Rose, sorry. A bacterial cell is a prokaryotic cell and a plant cell is a eukaryotic cell. But we've kind of covered that here, haven't we? That's what this means. Bacterial cell has no nucleus because it's prokaryotic, whereas a plant cell has a nucleus because it's eukaryotic. And just to repeat before we move on, a flagellum is like a tail. It's used for movement, and a slime capsule is on the outside of some bacterial cells, and it's used to kind of help protect them from your immune system. What is chitin? Chitin is fungal cell walls. Fungal cell walls are made of chitin. Could you talk about pili? Again, you'd have to say, Ayush, that bacterial cells may have pili, plant cells don't. Because not all bacterial cells have pili. Tanisha, chitin is what fungal cell walls are made from. Okay, guys, I'm going to move on. Now, we are going quickly, but obviously we want to do as much revision as possible. Remember, you can rewind and watch this back if you didn't get all the answers down. This is a practical-based question, and I've chosen this required practical because it could come up, right? I'm not saying it will, but there hasn't been a question on the microscope required practical for a while. So a microscope has a ×5 eyepiece lens. Now the eyepiece lens is obviously the one that you look down. Let me just label it on here. So this is the eyepiece lens. Yeah, so that's ×5. Describe, nice and easy command word there, how to use this microscope to observe a prepared slide. And we're going to want a magnification of ×50. Okay, now, it's AQA, it's AQA. Separate and combined, Jelly, separate and combined. Good spicy. Right, Tilly, I'm going to just pull you up on that. I'm glad you said it. I was hoping someone would. You don't have to talk about making the slide in this question because, Tilly, they've given you a prepared slide. So it's already made, right? That's why this is only 4 marks. They've already made the slide. So they've already got the root hair cells, they've already put them on the slide, they've already stained them, perhaps with iodine, like you said, they've already stained them, right? So read the question carefully. It's already prepared. So we're starting with a prepared slide. We just need to go through the steps of actually viewing them on the microscope. So what would we say? Put the slide on the stage. Give me some more suggestions in the chat, please. How do you prepare a slide though? Well, remember, you want the thinnest layer of cells possible, and you want to add a stain so you can actually see the cells. Like potentially you could say add iodine to stain the cells. Put the slide on the stage. You could also say, which I don't think anyone has said, turn on the light or the lamp, because most microscopes have a light, right, that comes up underneath. So you could have got a mark for that. You didn't have to. Yeah, you do start, a lot of you are saying in the chat, start with the lowest magnification or the lowest power objective lens. Because if you have a look at a microscope, can you see these lenses here? These are called the objective lenses. And you usually have like 3 or 4, and you do want to start with the lowest magnification or the lowest power objective lens because it makes it much easier to focus. Okay. Can't say lowest resolution though, Isabel, because microscopes have a set resolution. Like this microscope will just have a set resolution, and it's probably going to be quite low because it's a light microscope. But you can change the magnification by changing the objective lens. Freddie, we've already prepared the slide, just so you know. You don't have to talk about putting a cover slip on the slide because it's already been prepared. Use the coarse focusing wheel or the coarse focusing knob to focus on the specimen. What else? Let's have a look at some of these answers. Oh, I like that. You've got clip it in place as well. Use the fine, yeah. Use the fine focusing wheel to improve the focus. Now I'm kind of writing down lots of possible answers here. It's only 4 marks. Why do you use the lowest magnification? It's the easiest one to focus on, and it gives you the widest field of view. So you always start with the lowest magnification. You get it into kind of rough focus using the coarse focusing wheel. And once you've got it kind of in focus, then you use the fine focus wheel to get it in perfect focus, if that makes sense. And then we need to think about our magnification. So well done. If you remembered in the question, it said we wanted a magnification of times 50. So then we're going to have to use the times 10 objective lens. Why do we want to use times 10? Excellent, Hannah Flo. Because the eyepiece lens was times 5. So we want to use the times 10 objective lens because that gives a total magnification of times 50. Yeah. You don't need to add the equation for this. Well, do you mean the equation that I've just kind of written? To work out the total magnification, you do times 5 times 10. Because total magnification of a microscope is the magnification of the eyepiece lens, which was times 5, multiplied by the magnification of the objective lens, which was times 10. So here we'd have a total magnification of times 50. Okay. Can you bullet point? Yes, you can. Yes, well done, Noam. That is how you work out the total magnification on a microscope. Which points are most important or best to write? Well, you needed to have at least one of these, okay? Because the question did specify that the magnification had to be times 50. So you needed to have at least one of these points specifically about the magnification and how you're going to make sure it's times 50. And then you could have had any three of these points. So easiest ones, put the slide on the stage, turn on the light, any of those. Can you give hazards and risks? You didn't have to here because it didn't ask you specifically, but obviously if it had asked you specifically, I mean, there's not many risks though with using a microscope, right? The coarse focusing wheel, yeah, it does. It moves the stage to get it into focus in Aya. Yes, it does. This could be foundation or higher, by the way. This topic, using a microscope, it's a required practical. It's on both foundation and higher, combined and separates. How do you prepare a slide? Let's just briefly, you want a single layer of cells because obviously you need the light to pass through, okay? We should know that. We want to be able to see the cells. So we need a single layer of cells so the light can pass through, and you're going to add a stain, and then you're going to put on a cover slip, which you lower slowly. You lower your cover slip slowly because you don't want air bubbles under there, right? Because they're going to affect how you see. How do you know times 10 objective lens? Well, the question said I needed a magnification of times 50. And my eyepiece lens was times 5. So I have to use a times 10 objective lens because times 5 multiplied by times 10 would give me 50. Okay, what would the risks be? I don't know. The cover slip is sharp. The slide is sharp. You might cut yourself. Suppose if you've used a stain, the stain might be like an irritant or something. So maybe you should wear gloves. Okay. You might blind yourself. What if if the stain gets in your eye, so wear goggles, maybe. Right, this is a question about the cell cycle. This is a maths question. Do you always stain with iodine? Not always, but for GCSE, that's usually what we use. If it was a different stain, they'd probably tell you in the question. The time taken for a complete cell cycle is 15 hours. Calculate how many hours, and we've got the command word calculate. So immediately we know we're using our calculator. It's a maths question. How many hours did the cell spend in mitosis? And give your answer to 3 significant figures. If you have a look, mitosis is 28 degrees. Out of the whole pie chart, which is 360 degrees. Oliver, well done. So mitosis, the angle is 28 degrees out of the entire pie chart, which is 360 degrees. And then you can multiply that by, oh, you don't need to multiply it by 100, do you? I was going to work it out as a percentage, but you don't actually have to. Let me see what I get, see if I get the same as you. Remember, you're giving your answer to 3 significant figures. So 28 divided by 360 times by 15 hours. Yes. Times it by the 15 hours. So we're working out a proportion of the cell cycle. Now my calculator says 1.16 recurring, which means 1.166666, right? So if I want to give it to 3 significant figures, that's going to be my final answer. I'm going to have to round up because I've got 6s there, which are bigger than 5s. So I'm going to round up and give it as 3 sig figs. Why do you times it by 15? Because it's that proportion, 28 out of 360, and the cell cycle is 15 hours. The way you could do it, guys, if your brain works a little bit better with percentages, like mine used to, I remember when I was doing my GCSEs, you could have times that by 100 and worked out what percentage of the cell cycle was spent in mitosis. And then that would be the percentage of the cell cycle. So you could have done it like that. You could have worked that out as a percentage. Does that make sense to anyone else? Or is that just how my brain works? You could have done it like that. And then you would have thought like, right, 7.7 recurring percent of the cell cycle is spent in mitosis. And then you could find 7.7 recurring percent of 15 hours. So you could have done it that way. Okay. But you want it in hours. Yeah, you do. So always check you're giving your answer in the right units as well. Because yeah, units are always important in biology. Okay. Describe one change in the cell that occurs during each of the stages. So we've got stage 1, stage 2, which we're told is mitosis, and stage 3, and we need one change. You should be able to give more, but we need at least one. Okay, excellent, Bella. DNA replicates. What else could we have said? Increase in the number of subcellular structures. You could have said that. I'm very impressed so far. We only needed one, but I'm just giving you options here. So increase in the number of subcellular structures, or you could have even named some, right? Increase in the number of mitochondria, increase in the number of ribosomes. But the big one we're all going to remember is during stage 1, we have to replicate the DNA because the cells are going to divide. So each daughter cell is going to need its own set of DNA, its own set of chromosomes. Okay. Stage 2, Mohammed says the nucleus divides. Yeah, that would be accepted. Or 2 nuclei form. Chromosomes move to each pole of the cell. Yes. The best way to phrase this for AQA is one set of chromosomes is pulled to each opposite pole. That's how it's worded on the AQA specification. Can you just say interphase for stage 1? For AQA, no, not for this question, because it said describe one change in the cell that occurs. It didn't ask you to name the stage. You are correct. It is called interphase, stage 1. So if you know that, well done. But you're not describing what happens then, are you? You're just naming it. But yeah, remember, we only need one thing that occurs. So for stage 2, either the nucleus divides or one set of chromosomes is pulled to each opposite pole. Stage 3, again, well done. Some of you know that stage 3 is called cytokinesis, but let's actually describe one change. What is happening? Cytoplasm divides, well done. What else could we say if we want an alternative? Let's imagine we're asked to give 2 things. Cell membrane divides. Excellent work. And yes, 2 daughter cells are formed. Now, because this is mitosis, are they going to be genetically different or are they going to be varied? Genetically different, or is there going to be genetic variation? Identical, yeah. So let's add that in because we know that as well. 2 genetically identical daughter cells, because remember, we replicated the DNA and then one set of chromosomes moved to each opposite pole. So the daughter cells will have their own set of chromosomes and each of those 2 daughter cells will be identical. Okay, now this is like one of the differences, right, between AQA and Edexcel. So just a heads up here, Edexcel, you have to know the detail of mitosis in way more detail than this. You go through all of the stages, don't you? You don't do that in AQA. So that would be something on your Edexcel specification that you would still have to revise. Do the number of subcellular structures double, or do they just increase randomly? I don't think they specifically double. I think they just increase in number. But you could also say things like mitochondria replicate or ribosomes replicate, or just they increase in number. Okay. Okay. DNA replicates is fine. Mariam, DNA replicates. All chromosomes replicate is fine. By the way, these would be diploid cells, not haploid. Diploid cells, because they have pairs of chromosomes. They have, basically what that means, diploid daughter cells, is they have a full set of chromosomes. Their chromosomes are still in pairs. You definitely don't have to write cytokinesis. You're not expected to actually know it's called cytokinesis, stage 3. So you definitely don't have to write that. Yeah, and well done, Umar. Meiosis makes haploid cells. Mitosis makes diploid cells with a full set of chromosomes because the chromosomes are still in pairs. The topics are kind of in order of the spec, to be honest. Like this is all module 1 for AQA, cells. Okay, cytokinesis is an A-level term, yeah. And this is still module 1, cells. So all we've got to do here is match up the diagram to the process. Now this could be foundation or higher, but it gives us an opportunity to also just write down what we know about each of these processes. So put your answers into the chat. Again, TRXYS. You wouldn't get a mark for just naming the stage for that previous question because it asked you to describe what happens. Okay, so water molecules, right? The clue here, we've got water. So it's got to be osmosis. It's got to be. 2 is diffusion. Well done. And we'll explain why in a second. And that means 3, or the magnesium ions, has got to be active transport. A boy, it depends on the concentration, right? If you've got a higher concentration of nitrate ions outside of the cell, they can diffuse in. If you've got a lower concentration of ions outside of the cell, then they would have to use active transport. So ions can use both diffusion and active transport. It just depends on the concentrations, right? So let's get some definitions down for these, I think, and put them into the chat if you think you know them. We want to make sure we get them right. I'll try and watch my handwriting. There we go. Oh, I forgot that. Through a partially permeable membrane. It's always sensible to add that on, isn't it? For osmosis, because sometimes that's a mark. But as soon as we see water in that first picture, we know it's osmosis, because osmosis is the movement of water from a dilute to a concentrated solution through a partially permeable membrane. Dilute to concentrated, that's one way you can say it, or the other acceptable answer is it's the movement of water from a higher concentration of water to a lower concentration of water. And you can see with this root hair cell, there's a much higher concentration of water outside the cell. So it's obviously going to move in because it's more dilute outside the cell than inside. So it's going from dilute to concentrated. Let's do the others. Diffusion. I'm looking at your answers actually. Well done. Yes, the movement of particles. Now this could be like in a gas or in a liquid. It could be, I'm just going to say particles or molecules or something, from a region of higher concentration to a region of lower. Now, does that always take place across a partially permeable membrane? Would I add that to my definition? Yeah, you can, boy. You can say water potential for osmosis. If you're going to say that, you'd say from a high water potential to a lower water potential. No, exactly. It doesn't have to be across a partially permeable membrane, does it? Because I could spray some air freshener over here and it could diffuse over there. So we don't add that to our definition because it doesn't have to include a membrane. Right, guys, in the chat for me, can you tell me what active transport is, please? Very important. Well done, Aaron. Well done. Aaron, because it's going against the concentration gradient, what does it require? Because we should include that. And the movement of particles, what should we say? Should we say against the concentration gradient? And it's really important that this requires energy because you're moving against the concentration gradient. The other way to phrase that is to say the particles are moving from a lower to a higher concentration. It's like you're going up a hill, right? From low to high. And if you go up a hill, it's hard work. It requires energy. Whereas diffusion is passive because diffusion is going from high to low, which I always think is like rolling down a hill. It's really easy to roll down a hill. It does not require energy. It's passive. Okay. Okay. Let's continue because some of you are asking me questions like Fahim has asked me about chlorosis. I know that that's coming up later. Okay. Okay. The questions do get harder, by the way, as well. The questions are going to get harder, and we're going to look at some separate science-only questions at the end as well. Right, so we've got a practical question here. This is like the Osmosis Practical. So students investigated the effect of different concentrations of salt solution on the mass of pieces of potato. Standard prac comes up a lot. They're cutting 3 pieces of potato. Each have a mass of 2 grams. They're placing the pieces of potato into a salt solution. After 30 minutes, they're measuring the mass of each piece of potato and calculating the change in mass. And then they're doing this for 5 other concentrations of salt solution. Guys, before we do the actual questions on the screen, can you tell me what is the independent variable? What is the independent variable? Anya knows the song. The independent variable, I've kind of given you a big hint here, haven't I? It's the one you change. So we're doing this in different concentrations of salt solution. Okay, look, they're here. These are all the different concentrations of salt solution. We've changed the concentration of salt solution on purpose to see if it affects the mass of a potato. So that's our IV, our independent variables. Now, then we need to think about control variables, which are things we keep the same. Yeah, because if we don't keep them the same, they could affect the rate of osmosis. They could affect the mass of the potato. So don't refer to mass or time. What other? Don't refer to mass or time because it told you the mass was controlled and it told you the time was controlled. So what else? We can't refer to mass or time. Right, temperature is a really good one because temperature for sure is a factor that affects the rate of osmosis. The type of potato, or you could even just say, use the same potato. I don't know. I don't even know any different types of potato, like a Maris Piper versus something else. Use the same potato. Pressure, I don't think would be accepted. Same volume, that would be a good one. It's not water though, is it? It's salt solution. So same volume of salt solution. So make sure you're actually using, yeah, it says salt solution. The size or the surface area of the potato, because it says that they've controlled the mass, but it hasn't said that actually they've used the same length or the same diameter. So you could say the same dimensions or the same size or the same surface area. Can't have mass, Iron, because it said do not refer to mass or time. So you can't have time either. So some of you, Evie, make sure you read your questions tomorrow really carefully so you don't repeat answers that they've already given and therefore are not going to get you the marks. And the other one you could say is make sure there's no skin on the potatoes, on the cylinders, because skin is like a barrier to osmosis. So for sure that would affect your results. Okay, yeah, no skin. Well done. Why keep temperature the same? Guys, temperature is so important, isn't it? If you increase the temperature, then obviously the water molecules would have more kinetic energy and then the rate of osmosis would be faster and that would certainly affect your results. Yeah, radius would be fine. Radius would be fine. I can't say mass or time because it says do not refer to them. Calculate value X. Guys, I'm not even going to do this. You're going to do it because I'm just checking that you can calculate a mean and I'm 100% certain that you can. I'm going to trust you. Ooh, why is it worth 2 marks? Why is it worth 2 marks? Have we got an anomaly? No, I think the second mark was for the minus sign. Yeah. No, they've included all the results in the other calculations, haven't they? So they've included all the results in the other calculations. So I don't think we're looking for an anomaly. But my hint would be if you're calculating a mean, which is a really straightforward calculation, if it's worth 2 marks, that means there's something else. So it's either going to be, is there a result that's way off and doesn't fit the pattern, in which case it's an anomaly, you don't include it. Here, if you look at the other answers, they have included all 3 of the results in their mean calculation, even when some of them are quite different. So it's not that they want you to discard an anomaly, it's you're getting a second mark because it's a minus, and you've got to put the minus basically in front of your answer. Yeah, Ash, I kind of thought is minus 0.26 an anomaly. But if you look at the other calculations, and again, this is like a hint for you to do in an exam, like here, you might think that minus 0.22 is an anomaly, but if you look, they have included that in their mean. And this first one, you might think that 0.25 is an anomaly, but they have included that in their mean. So you can include this in your mean, but the second mark is for that minus sign, okay? An anomaly is just a result that does not fit the pattern. It doesn't fit with the other results. It's like too far away from the other results. And sometimes what we do is we don't count it when we calculate our mean. We ignore it or discard it when we calculate our mean. Okay, the one you measure is the dependent variable. Yes. So in this case, it's the mean change in mass is our dependent variable. Okay, the mean change in mass. Do you want me to just label it on here? This one here, that's our dependent variable, the one that we measure. So you should know your independents, your controls, and your dependent variables. Why are potatoes losing mass? Osmosis. If they're losing mass, water has been moving out by osmosis, right? Across that partially permeable membrane. How do you calculate a mean? You add the results together and divide by the number of results. So in that previous question, we'd have to add the 3 results together and then divide by 3. Sorry, I just assumed that everyone put an answer in the chat, so I assumed we all knew that. But yeah, you add your 3 results together in the table and then divide by 3. Right, this one is an osmosis question. It's quite a hard question, this one, so I'm guessing this would be a higher tier question. Although, having said that, you could get a similar question on a foundation paper because osmosis is on both. Right, the figure shows a potato cell. Explain why the mass of the pieces of potato increased in a 0.2 molar salt solution. And what makes this hard and weird is it says you should refer to the cell parts labeled in the figure. So we know it's increasing in mass, so that kind of gives us a clue. Like, that makes it a little bit easier. It's increasing in mass. Why? This is AQA. Let's start with the easy stuff. Okay, yeah, so water moves in by osmosis. Now, you might be thinking, how did I know that? Well, it's easy. If it's increased in mass, why is it increased in mass? It must have gained water. So water has moved in by osmosis. We can all get that mark. Now we have to talk about, because it told us in the question, refer to the cell parts in the diagram. So water moves, first of all, across the cell wall. Why? I always bullet point every answer because the cell wall is permeable. It's fully permeable, the cell wall. Yeah, Debola, you're getting like a mark for each structure. It's a bit of a weird question, isn't it? But the cell wall is permeable. And then water moves across the, what else is labeled? The cell membrane. Why? Because it's partially permeable. So it's not fully permeable, it's partially permeable. We know that about cell membranes, but it does allow water through. Water is one of the molecules that can pass the cell membrane. And then finally, water moves into the cytoplasm. Okay, and obviously the cytoplasm is going to fill up with water, right? Now we've still only got 4 marks. What else do you think we need to say? Yes, semi-permeable membrane or partially permeable membrane is the cell membrane. It's better to say partially permeable than semi-permeable. This is 5 marks. Selectively permeable is okay as well. What have we not actually said? We've not explained why the water moves in. So let's see if we can do that. Water moves in because, and I'm going to word it like this, and you can tell me whether it makes sense to you. The solution outside the potato is more dilute than the solution inside the potato or inside the cells. Let me check your other answers. The concentration is lower inside the cell. That would be fine as well, Abby, because I'm assuming you're talking about the concentration of solute. There's a lower concentration of water inside the potato than outside. That would be fine as well. It always causes confusion because different teachers teach different definitions of osmosis. So some of you will be saying it moves from a dilute to a concentrated solution. Some of you will be saying it moves from a higher concentration of water to a lower concentration of water. But that's the same thing because a dilute solution, like out here, it's more dilute, right? A dilute solution has a higher concentration of water, doesn't it? Think about your orange squash. If it's dilute, there's more water in it. So you can either say it goes from a dilute to a more concentrated solution, or it moves from a higher concentration of water to a lower concentration of water. It's the same thing. Could you say it becomes turgid? I mean, it would, yes. It wasn't actually worth a mark, but it would because it's a plant cell. I know it's a plant cell because it has a cell wall. So the cell becomes turgid. It won't burst, will it? Because the cell wall stops it from bursting. And I suppose there might be like a follow-up question on that. The cell wall can like withstand the pressure. We actually call it turgor pressure, which I saw someone say in the chat before. So the cytoplasm is like filling up with water. You've got this pressure, but because this is a plant cell, it won't burst because of the strong cellulose cell wall. The cell just becomes turgid. So turgid, Harvey, is when the cytoplasm is full of water and it's like exerting a pressure on the cell wall, but the cell wall is so strong that the cell does not burst. Osmosis is only for water, yes. Semi-permeable is okay at GCSE, it's just not for A-level. So I always use partially permeable for GCSE and A-level. But yeah, partially permeable, selectively permeable, semi-permeable. From lower to higher, yeah, if you're talking about solute, Freya, if you're talking about solute, it's moving from a lower concentration of solute to a higher concentration of solute. But if you're talking about water, it's moving from a higher concentration of water to a lower concentration of water. So you've got to be really, really careful with osmosis definitions, which is why I always just say the water moves from where it's more dilute to where it's more concentrated. Okay. The opposite of turgid is plasmolyzed. So plasmolyzed plant cells have lost a lot of water by osmosis. And yeah, you can say water moves from a high water potential to a low water potential. There's so many different ways of saying it. Flaccid is, yeah, flaccid is when it's lost some water. Plasmolyzed is like extreme flaccid. Yeah, plasmolyzed is it's lost a lot of water. Flaccid is like it's lost a bit of water. Okay, right, let's carry on. Hypertonic. Hypertonic means it's got a lot of solute, a high concentration of solute. I always think of hyper, like hyper to me is like lots of sugar. So a solution is hypertonic if it's got like a high concentration of sugar or salt. Right, the image in the figure was made using an electron microscope, not a basic light microscope like you have in school. Give one piece of evidence to support this. Ooh, Joe says it's black and white. Joe, you cannot say that because your whole exam paper is probably black and white, isn't it? Because they don't want to pay for colour copying. So your exam paper images will be black and white anyway, yeah? Or at least they always have been. So I wouldn't go for that one, no. But you could go for, it has a high resolution. Well done if you said that. You could say you can see small subcellular structures, as we call them at GCSE. High magnification, the mark is always for resolution. So you can say it has a high resolution and magnification. But you must say resolution because that's what will be on the mark scheme. If you just say high magnification, you probably won't get the mark. If you just say more detail, I don't think you get the mark, but you could say can see more detail of subcellular structures, which just means a structure inside the cell. And obviously that's because it's got a high resolution and a high magnification. You can see the subcellular structures, you can see detail of the subcellular structures. And yes, Mimi, the other one you could have, we only need one, but you could say it's 3D. Now, my question for you, if it's 3D, what type of electron microscope have we used? Yes, it is, Umar, well done. If you wrote two answers and one was right but one was wrong, no, you'd lose a mark. That's the risk, isn't it? It's called the list rule. If you give two answers because you're not sure, if one's right and one's wrong, they will take away the first answer. If they're both right, it's fine. Yeah, so interestingly, because this is 3D, it's actually a scanning electron microscope because they give 3D images. Now, you didn't have to say scanning, but it must be an electron microscope. In fact, it must be a scanning electron microscope because it's clearly 3D. Resolution and resolving power are the same thing. Yeah, you should know the different types of microscope. Light microscopes obviously have lower magnification, lower resolution. But the benefit of a light microscope is that you can see colour and you can look at living specimens. Whereas with your electron microscopes, they're always black and white and you can only look at dead specimens. Because you're putting them in a vacuum because you're using electrons, right? So you can only look at dead things, but electron microscopes have a much higher magnification and a much higher resolution. Right, let's do a maths question. There's always something on calculating magnification or calculating real size, for example. The potato cell in the figure above contains starch grains. Starch is obviously a store of glucose. A starch grain on a different image, so you don't have to use that picture. So don't start panicking and getting your ruler out and measuring that picture. Read the questions carefully, guys. Different image had a diameter of 1.2 centimeters. The starch grain had a real diameter of 0.008. Can you work it out for me? Yeah, Chloe, that's correct. First thing I would do, I can see some correct answers already, but just to go through exam technique, immediately I would convert this into millimeters because I can see that they've given me the real diameter in millimeters and I can't do my calculation if my units are not the same. So 1.2 centimeters is 12 millimeters. Do we all understand that? This is because 1 centimeter equals 10 millimeters, right? So if I'm going from centimeter to millimeter, I clearly have to multiply by 10. And then there's no harm, don't be embarrassed, I still do it. There's no harm in just doing a little sketch of your formula triangle. Beware though, you won't get credit. They're not going to give you a mark for the formula triangle. Yeah, I'll do some more conversions actually in a second. They're not going to give you a mark for the formula triangle. So I would also write it out, okay? Don't just rely on the triangle. Magnification equals image divided by actual. Because if you write it out, they then give you a mark, okay? Then the image we've got in millimeters now. So image was 12 millimeters, actual. So the magnification is that, isn't it? I think. I mean, I just copied your answers in the chat, but I was assuming you were all right. 12 millimeters divided by 0.008 millimeters. Yeah, thanks, guys. Right, a few of you have asked me to do some other conversions. So I'll do you a quick little diagram, which you can copy down, which might be helpful. I'll do the common conversions that you need for biology, okay? So meters to millimeters to micrometers to nanometers. These are the ones on the AQA specification that it says you should be able to do. So basically, if you're going in this direction, you're always going to times by 1,000. So it's nice and easy. So what you could even do in your exam, if you're not confident with your conversions, is write that down. Just write down meters, millimeters, micrometers, nanometers, each time you're timesing by 1,000, right? And if you're going backwards, it's divide by 1,000. Okay. Do we convert to millimeters or micrometers? For magnification, it doesn't matter which unit spell as long as they're in the same units. Okay, so like here, I had 1 in centimeters and 1 in millimeters. That won't work. So I converted the centimeters into millimeters, but I could have converted the millimeters into centimeters as long as they're in the same units. So if you do have to do centimeters, I think I've already done that one down here. So centimeters to millimeters would be times 10. And if you're going back the other way, it would be divide by 10. Okay, but we don't use centimeters that much. This is quite an unusual question. So centimeters to micrometers, well, think about it. You should be able to do that now. If I want to go from centimeters to millimeters, I have to times by 10. And then I know from my diagram at the top that if I'm going from millimeters to micrometers, I have to times by 1,000. So if you did have to go from centimeters all the way to micrometers, you could do it in 2 stages, or you could just multiply by 10,000, right? Okay, I'm not going through natural selection today because that's actually a paper 2 topic for AQA. Centimeters to meters. Well, again, you should be able to do that now, no? Centimeters to meters. Because there are 10 millimeters in a centimeter. And remember, there are 100 centimeters in a meter. I don't think you'll be asked that one, to be honest. That's quite a strange one. Meters to nanometers. Look, the diagram at the top is everything you need, isn't it? If you're going all the way from meters to nanometers, you're timesing it by 1,000 3 times. You could even do it in stages. If you're not confident, times it by 1,000 to get it into millimeters, times it by 1,000 again to get it into micrometers, and then times it by 1,000 again to get it into nanometers. Okay. Okay, but the one at the top, that little flow diagram at the top, they're the most common conversions for biology. Okay. I wasn't going to do food tests today because there was a big question on it last year. Big question on food tests last year. I mean, if you're really struggling, maybe we could quickly just jot them down at the end, but we'll see how we are for time. Meiosis is paper 2, so we're not doing that tonight. Right, this slide, I've not given you a question, which you probably notice. But I want to know from you what happens in the small intestine, and we're going to make some notes on how the small intestine is adapted for this process. Okay, so what happens first of all? What happens? Can you give us the 2025 papers? Unfortunately not. Right, so in the small intestine, let's write this at the top. It's the absorption of nutrients. Can you give me some examples of nutrients? What nutrients? What are the end products of digestion? Glucose or sugars? Some of you are naming enzymes, but they're not nutrients. Amino acids. What else gets absorbed here? Glycerol and fatty acids. Okay, so now we know what's going on here. We're absorbing these nutrients, which are the end products of digestion, and we're absorbing them into the bloodstream. And I'm going to add on to this diagram. In here, there are absolutely loads of capillaries, right? So these are my capillary networks. Let's just label that. So we're absorbing nutrients, which are inside our small intestine, into our bloodstream. So what are the adaptations? Let's have a look. I've missed loads. I'm sure you've all put loads. Right. So we could say there's quite a few we could talk about, but the main ones, the small intestine has villi and microvilli to increase the surface area for faster or more absorption. Okay, so notice what I've done there. I've not just said large surface area. I've said there are villi, which are the big folds, and there are microvilli. So you've got a huge surface area, so you get more absorption. 1 cell thick. What is 1 cell thick? Diffusion. Yes, so it could be diffusion or it could be active transport, depending on the concentrations, obviously. It's diffusion if they're going from high to low. It's active transport if they're going from low to high, which we've kind of covered already. Right, the wall of the villi are 1 cell thick. So you could link that to a short diffusion pathway. What else are we saying? Good blood supply. Or sometimes we say it like a dense network of capillaries. But that basically just means lots of capillaries, right? I've kind of drawn them on in red. There's loads of capillaries in each villus. And well done if you said that. It maintains the concentration gradient because, just let me explain that for 1 second, as soon as the nutrients get absorbed, the blood carries them away. So you're maintaining a concentration gradient, hopefully. There's going to be a concentration gradient so they keep being absorbed because the blood is going to carry those nutrients away. Oh, that's a good one, isn't it? It's very long. The small intestine. Now, you might think that wouldn't get you a mark, but if it's like a 4 to 6 marker, it does. The small intestine is very long, so more time for absorption. Notice that I'm trying to explain each adaptation. I'm imagining this is like a 4 to 6 mark question about how the small intestine is adapted for absorption. So you could say the small intestine is very long. I think it's about 9 meters long, so more time for absorption. Now, Alex, you read my mind. I was just about to say, Alex, that in here, there's lots of one particular organelle, and I was going to ask if you knew why. So in these cells that line the villi, there are lots of mitochondria. And I was going to ask if you knew why. Based on like methods of absorption. Yay, I saw that, Umar, well done. Cells have many mitochondria, so more energy released for active transport. So if you do have like a low concentration of glucose in your small intestine, you still want to get it into your blood, right? You're going to have to move it against its concentration gradient. You're going to have to use active transport. Active transport requires energy. Where does the energy come from? Respiration. Aerobic respiration takes place in the mitochondria. In fact, let's just remind ourselves the mitochondria is the site of aerobic respiration. Okay. What cells? So these cells here, can you see where this circle is? These are the cells here that line the villi, or the cells that make up the wall of the villus. So there's like a layer of cells that make up the wall of the villus. Surface cells, they're called epithelial cells, but you don't have to call them epithelial cells. But those surface cells contain lots of mitochondria to release lots of energy, more active transport. Yeah, the projections, boy, the projections are called the villi, but then on the villi, you've also got microvilli. So you've basically got a huge surface area, faster absorption, and the wall is only 1 cell thick, short diffusion pathway. You've got all these different adaptations. Anaerobic respiration is in the cytoplasm, by the way, Lee. Yeah, these are, I don't want to call them predicted topics, Fatima, but based on my analysis of the last like 6 years of papers, these are things that either haven't come up for a while, didn't come up last year, things that potentially could come up, like the lungs and the alveoli came up last year, so maybe the small intestine could come up this year. Could you also say the external medium gradient is also maintained? The concentration gradient is maintained, Mariam. The concentration gradient is maintained. What is 1 cell thick? The wall of the villus, or the walls of the villi are 1 cell thick. The surface layer is 1 cell thick. This is higher, but it's also on foundation. I mean, if it's a big 6 marker, it's more likely to be on higher, but this content is also on foundation. Okay, you try writing this quickly and reading the chat at the same time. See what your handwriting is like. Right, let's get some labels on. What we're going to do here is quickly label the structures in the digestive system, but then we're also going to annotate it with where the enzymes are produced and what enzymes. Okay. Yeah, some water is absorbed in the small intestine and then more water is absorbed in the large intestine. Right, so get your answers into the chat. Let's just label it for now. This is the digestive system. We're moving on to like digestive system and enzymes. We'll label it and then we'll go through the different enzymes where they're produced. Okay. How nice would it be to just get a question asking you to label? Yeah, the salivary glands are in the mouth. Do you want me to add those on as well? Because that's important because that's where the enzymes are produced. Stomach, someone said contains hydrochloric acid. Well done. E is the liver. Well done. D is the pancreas. Well done if you're getting these right. I can see lots of you are. F is the small intestine. I mean, I could label the large intestine, but we don't really learn anything about that for GCSE AQA, do we? But we could label it. Yeah, this is separates and combined, guys. The first 3 hours, everything is separates and combined. Everything. Yeah, lots of water is absorbed in the small intestine. Ooh, E, the bile is produced. Right, let's go ahead now. I might just change my pen colour. And let's add on which enzymes are produced because that's what's most important. So the salivary glands produce amylase. Or you could, I don't know whether you guys just call it carbohydrates because you can get away with that for GCSE. What about the stomach? Oh, and by the way, don't just say the mouth produces amylase. It's the salivary glands in the mouth, okay? Be specific. Right, the stomach produces protease. Excellent. Breaks down proteins. I'm going to show you the job of the enzymes in a second. Don't need to know the different types of protease for AQA. You just need to say protease. No, Megan. Salivary glands, it's only amylase or only carbohydrates. Right, what about the pancreas? The bile is not an enzyme. I'll add that on in a second, honey, but bile is not an enzyme, by the way. Right, pancreas produces all protease, amylase, lipase. I just remember it's all three. The pancreas is just all three. The one that breaks down protein, the one that breaks down starch, the one that breaks down lipids. All three. Small intestine, again, all three. So it should be relatively easy to remember. Small intestine. Yeah. So when you're learning this, right, salivary glands, just amylase, stomach, just protease, and then you can just lump it together and say the pancreas and the small intestine produce all three of the main groups of enzymes, protease, amylase, and lipase. Now, loads of you are talking about the liver. The liver does produce bile. But just so you know, bile is not an enzyme. Don't talk about it in an enzyme question. If they specifically ask you about the role of bile, we've got a question coming up later on that. So I'm going to wait to talk to you about bile later on. But yes, it emulsifies lipids. Well done. It produces bile, which is then stored in the gallbladder. Good knowledge from lots of you there. So the liver makes the bile, it's stored in the gallbladder, and then it's released into the small intestine. Yeah, amylase is a carbohydrate, Lex. So at GCSE, you can say amylase or carbohydrates for AQA. It is fine. Okay, what do the enzymes break down? You have read my mind. Let's have a look. Enzymes in digestion, right. Again, not a question. This is just revision of the key enzymes, what they break down and what they break it down into. But I do also want to add, like, the whole point of these enzymes, and I'd like you to write this down if you're making notes, is to break down large, insoluble molecules into small soluble molecules. Why? Guys, why in the chat? Thinking about digestion, why do we need to break down large insoluble molecules into small soluble molecules? Like, why do we need to break down starch into glucose? Exactly. So not so it's easier to digest, it's so we can absorb. We've literally just done the small intestine, right? In the small intestine, we absorb nutrients. We cannot absorb starch, protein, lipids. They're too big. So we need to break them down into small soluble molecules. And I'm going to add on so they can be absorbed, as in they can go into the bloodstream. Yeah. Right. Yeah, in the small intestine. Right, so what do we need to know? Let's just kind of circle the enzymes. So carbohydrates, or you can call it amylase, digests starch, which is large and insoluble, into sugar. What type of sugar? Because we can be a little bit better than that, can't we? What type of sugar? Simple sugar, yeah. Glucose. Glucose, which is just a simple monomer, right? It's small, it can be absorbed. Then we've got protease, which digests protein, which is large and insoluble, into amino acids. What is maltose? You don't have to know about maltose for GCSE. That's on the AQA A-level specification. However, if you're Edexcel, that may be different. You might have learned that it's 2 steps. It goes from starch to maltose, then glucose. But for AQA, we can just say it goes from starch to glucose. Fructose is a sugar found in fruits, which is very sweet, very, very sweet. So we often add it to foods because you don't need to use as much of it to get the very sweet taste. That's fructose. So protein is broken down into amino acids. We all good with that? We all know that protein goes to amino acids. No, maltose is not wrong, Angie. You could say that carbohydrates digest starch into maltose, but for AQA, we say glucose. Yeah, we're all good with that. Right, then fats. Can we say lipids, actually? Lipids. They're broken down by, we've got lipase enzymes, and they're digesting the lipids, which are large and insoluble, into fatty acids and glycerol, okay? So what are we learning on this page? We're learning the name of the enzyme, what it breaks down, so the enzyme substrate, if you like, and the products of digestion that can then be absorbed. And this is something that we should definitely know. It could be a multiple choice question, it could be a table, it could just be name the product from protein digestion, name the enzyme. Substrate is what reacts. Substrate is like the reactant in an enzyme controlled reaction. Yeah, this live is recorded, so you can watch it again tomorrow morning if you want, or you can just watch bits of it back, whichever bits you want to go. Is it 1 glycerol to 3 fatty acids? I mean, not really. It's actually a monoglyceride and 2 fatty acids, but you can just say glycerol and fatty acids for GCSE. Again, it's like the difference between A-level and GCSE. We obviously simplify it, don't we, for GCSE? So we say lipids are broken down or digested into fatty acids and glycerol. You don't have to know the ratio, at least not for AQA. No. What is glycerol? It's a type of lipid. You don't have to know the structure or anything. It's just the product of digestion. Yeah, we're going to have a break in about 15 minutes. I'm sure we can do 15 more minutes. Enzymes, my favorite topic in GCSE biology. Right, guys, hit me in the chat. What is an enzyme? What is it made from? What do they do? What do we know? We can see the lock and key model here, can't we? But let's just do some wider revision notes. It's a catalyst. It's a biological catalyst because it speeds up reactions in living things. What are they made from? Protein. So they're made from amino acids, right? They're proteins. They lower, do you use this phrase in chemistry? They lower the activation energy. So less energy is required for a reaction to take place. Well done, Arya. Yeah, so basically in biology, think about all the chemical reactions that are happening in your body, inside your cells. They use enzymes. Enzymes speed up. Let's write that down as well. They speed up chemical reactions. Now, we've just looked at 3. We've looked at lipase, amylase, and protease. They're all enzymes involved in digestion. Let's have a look at our lock and key model. What is this model telling us about an enzyme? Fahim, they have a complementary shape. Which bit of the enzyme, Fahim, is complementary to the substrate? The substrate is like the reactant, so it could be starch, for example. Right, the active site is complementary to the substrate. Yeah, so it binds to the substrate, which you can see here. So substrate binds to active site. What do we call it? What do we call this here when the substrate is bound to the active site? The substrate is just like the reactant. So if this enzyme was amylase, the substrate would be starch because starch binds to the enzyme's active site. Both enzymes can break down things, they can build up things, they can break bonds, they can make bonds. Enzyme substrate complex, yeah. So when the substrate binds to the active site, we call it the enzyme substrate complex. Yes, and then at the end, what we need to realize is the enzyme is not used up, so can be reused. Yes, well done. Yes, these are my predictions. Don't know whether they mean anything, but these are topics that I think could come up. Right, so this is called the lock and key model. It's the idea that the active site is complementary to the substrate. And I should say as well, the enzyme is specific to one substrate, which is kind of why we call it a lock and a key, right? A key can only fit into one lock. It can only open one lock. So it's specific to a particular substrate because the substrate binds to the active site. That forms the enzyme substrate complex. And if when the reaction occurs, the enzyme is not used up, it's not changed, so it can be used again. Okay, what about the induced fit model? Again, that's A-level biology for AQA. So not needed for GCSE. We haven't done the circulatory system yet. No, we have not. Don't need to know about induced fit for AQA. Right. What is this picture showing me, guys? We're going to use this as an opportunity to talk about what can happen. So if you look down at the bottom of my screen, right, denatured. In this example, what has denatured the enzyme? Have a look. Enzyme is denatured, so the active site has changed shape. Right, we've got heat. So in this example, we've got high temperature. And you should know that high temperatures denature enzymes. We would need to use the keyword, the enzyme is denatured. Don't say killed, it's not alive, it's just a protein, right? It's not a living thing. So the enzyme is denatured, the active site changes shape. If the active site changes shape, what's the effect of that? What's going to happen now? The active site has changed shape. So yeah, 37 is optimum for our enzymes because that's our body temperature, right? Yeah, excellent. Substrate cannot bind to active site. Or if you're using key terminology, you could say enzyme substrate complexes cannot form. Yeah, you can. Enzyme substrate complexes cannot form. You could say the active site is no longer complementary. The reaction can no longer take place. Excellent. Now, remember as well, extreme pH can also denature enzymes. So if the pH is too high, like way above optimum, if the pH is too low, way below optimum, that can denature enzymes as well. You say exactly the same thing. The enzyme is denatured, the active site changes shape, the substrate cannot bind to the active site, so the reaction will stop. The enzyme cannot work. Okay. You can say the active site binds to the substrate or the substrate binds to the active site. It doesn't really matter. Do we need to know the pH? I mean, the pH in the body is slightly different. Like the pH in your mouth, your saliva is about pH 7. In your stomach, it's about pH 2, 1.5 to 2. And then in your small intestine, it's about pH 7, 7.5. But any enzyme, if you take it away from its optimum pH, if it's too high or too low, it could denature. Do low temperatures denature? We've done some active transport already. Do low temperatures denature? Just checking. Right, so let's just make a note, because a lot of students make this mistake. Low temps do not denature. All it means, if you've got a low temp, it just means there's less kinetic energy. So be careful, yes. It just means the enzyme and the substrate have less kinetic energy, so you get less, what's the word I'm looking for? Less frequent collisions. They don't collide as often. You get fewer successful collisions because there's less kinetic energy. So it would reduce the rate of reaction, but it's not going to denature an enzyme if it's just a low temperature. Well done. Yeah, less kinetic energy. What we'd say in an exam is the enzyme and the substrate have less kinetic energy, so less successful collisions or less frequent collisions, so the rate of reaction is lower. Okay, cool. Do the amino acids in the enzyme break? The bonds, MH, again, that's creeping into A level, but when we denature an enzyme, the bonds between the amino acids break. So the active site changes shape, yes. Yeah. But for our enzymes, our body temp is 37. So 37 is the optimum temp for our enzymes. But do remember that different species have different enzymes with different optimum temperatures. So in a human, optimum temperature is 37, but you might get, I don't know, like an enzyme from a bacteria, and the optimum temperature might be 70 degrees. Right, complete the table. We're going to carry on for 10 more minutes, guys, I think. Complete the table to give one site where these digestive substances are made. We've kind of done this already, so you should remember. Bile is made in the liver. Excellent. What about amylase? Mouth. Oh, no, we can't say mouth. Can we? Because it's not produced by the mouth. It's in the mouth, but it's produced by the salivary glands. Lipase, we just need one site of production. So we could say pancreas, or we could say small intestine. What about protease? Again, we could say either, couldn't we? Because remember, the pancreas and the small intestine produce all three. So in fact, for amylase, we could have said pancreas or small intestine as well, couldn't we? I think some of you have maybe put that in the chat. Yeah, for sure, because pancreas and small intestine produce all three. So that's easy to remember. But the stomach is just protease. And remember, the stomach has hydrochloric acid, which gives the stomach about a pH of 1.5 to 2. It's very, very acidic. And then the salivary glands produce amylase. Yeah, I keep missing what, there's so many options that I've missed off stomach for protease. Yeah. Right, describe 2 ways that the mouth can break down starchy foods. Specialized cells is on paper 1, yes, but nerve cells comes up again in paper 2. The active site of the enzyme is the specific bit of the enzyme where the substrate binds. So the active site is the bit that's complementary to the substrate. Right, so describe 2 ways. So some people have said chewing teeth to break down, I don't know, how should we describe this? Large chunks into smaller chunks. That's called mechanical digestion. So you could have said for that one, mechanical digestion. Because it's not just enzymes, right? Enzymes would be chemical digestion, but we also have mechanical digestion in the mouth. We've got our teeth, we've got our tongue, we've got our chewing. That's breaking down large chunks into smaller chunks. But then we've also got amylase, or carbohydrates, as you can call it, which breaks down starch into glucose. That would be our chemical digestion, because that uses enzymes, yeah, which are proteins. Okay. No, it just says describe, doesn't it? So I actually don't think you do have to explain it. Could you say churning the food? Yeah, you could, because your tongue, which I always say tongue and people take the mic, but your tongue like moves the food around and kind of churns it to help break it down. Same thing happens in your stomach. Your stomach is made of muscle, right? So your stomach churns the food as well. Right, how does the liver help digest fats? Now, we mentioned this before, but let's see if we can get a perfect answer. Mechanical digestion and chemical digestion. Yeah, you could for that one, because it didn't say explain. Okay, so the liver makes bile, which, and this is a key phrase we should be using, emulsifies fats or lipids. Now, what does that mean? What would our second marking point be? I've seen some really good answers. We'll talk about the neutralizer stomach acid as well. So what we need to say, when we say it emulsifies fats or lipids, it increases the surface area. So lipase activity is faster, because you're basically going from big globules of fat, you break them down into smaller globules. This is called emulsification. And that's what bile does. It breaks down the big globules into smaller globules. We say it emulsifies the fats, but then it's important to remember that the reason for that is you're increasing the surface area so that lipase can then act faster. Lipase enzyme has then got more surface area that it can get to, so lipase will then be able to digest it faster. Okay. What are globules? Like big spheres of fat. Yeah, fat is like, it repels water, doesn't it? You get these big globules or big spheres, and it's very hard for lipase to digest it. But because bile emulsifies the fats, it increases the surface area, so lipase can act faster. Yes, Pranav. The other thing to just note, which some of you said in the chat, is bile also neutralizes stomach acid, which is really important, because think about those poor enzymes in the small intestine. You've got all this acid coming out of the stomach that could potentially denature the enzymes in your small intestine. But it's okay, because bile is alkaline. So bile neutralizes the stomach acid so that you get the optimum pH in the small intestine. Bile it's made by the liver, it's stored in the gallbladder, and it's released into the small intestine. So as it goes into the small intestine, it neutralizes the stomach acid, basically so you get the right pH in the small intestine. You get the optimum pH. Optimum means best, right? The optimum pH for enzymes in the small intestine. The meaning of emulsification is here. It increases the surface area of fats, increases the surface area of lipids, so lipase activity is faster. I mean, that is a harder question, by the way. That would probably be a higher tier question, wouldn't it? But for sure, we're going to do some really hard ones at the end. Bile is not an enzyme. No, it's an alkaline liquid, but it's not an enzyme. But it helps the lipase. It helps the lipase, doesn't it? by increasing the surface area of the lipids so the lipase can act faster. Right, we are going to do this so quickly because this is hopefully super easy. We're going to label the respiratory system. Yeah, ever so slightly alkaline, Grace. The small intestine is about 7.5, so it's only slightly alkaline. But yeah, bile definitely neutralizes the stomach acid. Super fast, love it. This is the main parts of your respiratory system that you should know. Yeah, so that's a bronchus, but remember you've got 2 bronchi, or you can call each one a bronchus. C are the smaller branches, which are called the bronchioles. D are the alveoli. Yes, for sure. And E, no, once they're denatured, by the way, it's the diaphragm, which is a muscle, isn't it? Okay. Now, what have I got here? I'm not going to spend any longer on that because that would like literally just be a labeling question and it would be super easy. I mean, I suppose I could add lung, but we know that, don't we? Right, over here is an alveolus. So it's a single air sac. Yeah, so you see all these alveoli that we had over on the left-hand side. This is just one alveolus. Now, what is the job of the alveolus? You've already told me in the chat. It is gas exchange. Yeah. How do the gases move in and out of the blood? How? You've got oxygen going into the blood, into the red blood cells, and you've got CO2 moving in the opposite direction. Yeah, it's by diffusion. So these gases are moving from a region of higher concentration to a region of lower concentration. That is the definition of diffusion. It will be passing across membranes, yes, because it has to go across the wall of the alveoli and the wall of the capillary as well. Yes, right. You're all preempting my next question. This came up last year, actually, so we don't know if it's going to come up this year, but give 3 features of the alveoli that allow large amounts of oxygen to enter the blood. And remember, that would be by diffusion. So you're kind of applying your knowledge of diffusion and how you would speed up diffusion. Oh, no, I'm not going to say that. Right, large surface area. And I always try and link it to the explanation. So if you've got a large surface area, faster diffusion or more diffusion. Right, thin walls. Again, link it. Short diffusion pathway. What else? Yeah, the walls are 1 cell thick. The wall is 1 cell thick. Moist wall never gets a mark, by the way. It's never on the mark scheme. Surrounded by many capillaries. How would we link that? You can actually see, can't you? You can see all the capillaries. Or good blood supply is fine. Good blood supply, rich blood supply, surrounded by lots of capillaries. Yeah, so that maintains the concentration gradient. And again, think about what that means. As soon as oxygen enters the blood, it gets carried away. So you're basically saying like there's always a lower concentration of oxygen in the blood than the air sac. There's always going to be a concentration gradient. So equilibrium will never be reached. Oxygen will keep diffusing into the blood. Well ventilated, yeah, the lungs are well ventilated. Again, that maintains the concentration gradient. I've got a question on that now in a second. Do you have to explain the concentration gradient? Not really, no. Maintains the concentration gradient. But the explanation is there's always going to be a lower concentration of oxygen in the blood than the air sac. So it will always diffuse from the alveolus into the blood. Moist walls is never on the mark scheme, just so you know. I mean, yes, they do have moist walls. Large surface area is always on there. That's a 3 marker. Name the process. We've already done this one. Don't know why I repeated it. Breathing allowed large amounts of oxygen. So someone said before that the alveoli are well ventilated. This question is about that. Breathing allows large amounts of oxygen to enter the blood. Explain how breathing does this. So breathing brings oxygen into the lungs or into the alveoli. Why does that allow large amounts of oxygen to enter the blood? We've kind of already said it, hasn't it? I would learn alveolus, Tiana. It brings oxygen into the lungs or into the alveoli, and it maintains the concentration gradient, or you could say there's a steep concentration gradient, which remember just means there's a big difference in concentration. If you keep breathing in and bringing oxygen in, what we're saying is there's always a higher concentration of oxygen in the air sacs than the blood. And diffusion is from higher to lower, right? So if there's a higher concentration of oxygen in the air sacs than the blood, it will keep diffusing from the air sac into the blood. So having that concentration gradient is super, super important. Inhalation, yeah, breathing in. Ventilation, inhalation, yeah, breathing in. Large surface area. For this question, yes, but not for this one, because this one's just about breathing. Not going to answer that question. What's the difference between respiration and breathing? Very, very different. Look, breathing is just this. Respiration is a chemical reaction, isn't it? In a cell that releases energy. Respiration is breaking down glucose like inside a cell to release energy. Okay, right. We are going to have a break. We're going to have, how long do you guys need? Can we just have like literally a quick 5-minute break? I just need long enough to nip to the toilet, get a drink of water, and then I will be back. You're all saying 15. Guys, if we have too long, we'll go over time. It's totally up to you. I can get, do you want to do 10 mins? Should we do 10 mins? Okay, fair dues. Right, can you make sure you're back here at 5? I'll write it on the, let's say 5:45. Okay? Yes, guys, 10 minutes. Just enough time for a toilet break and a drink, maybe a snack. And then we'll crack on. We've got another hour and a half to do. Thanks, guys. See you in a little minute. Hello, I'm back just to answer some questions. Yes, this will be left up on my YouTube. So if you want to watch it like later, or you want to watch certain bits back, or you want to watch it in the morning, you can do. What is meristem? Meristem are stem cells in a plant found like the tips of the roots, the tips of the shoots. They're the stem cells, right? That can differentiate into any type of cell. Did I scare you? The next bit is for triple and combined. We're still working our way through the content that is on both papers. So yeah. Yeah, we're going to do a question on monoclonal antibodies at the end. Auxins is paper 2. Meristems could come up on paper 1 though, because they're stem cells, right? And cells is on paper 1. I think I've got a couple of questions on stem cells coming up. Does osmosis require energy? No, Annie, it's passive, like diffusion. It does not require energy. Okay. Yeah, if we've got time at the end, I might do a little enzyme prac overview as well, I think. Right, guys, we're moving on to the heart structure. Is the mitochondria the powerhouse of the cell? No, we're not going to say that. We're going to say the mitochondria is the site of aerobic respiration, where energy is released. That's much better. Right, let's label the chambers first. Thanks officially, Yara. Let's label the chambers first, please. This should be really quick. Remember that when you look at the heart on a piece of paper, you have to flip reverse the sides because you're imagining it's like in your body, right? So don't get the sides mixed up. A is actually the right atrium. It's the left on paper, but if you pick that heart up and put it in your body, that would be the right side. B is the right ventricle. C is the left atrium, and D is the left ventricle. Okay, now tell me, which side deals with deoxygenated blood? Which side is deoxygenated, as in it's sending the blood to the lungs? Right, well done. Nearly everyone got that right. So the right is deoxygenated because it's coming into the right deoxygenated and it's going to the lungs. The left is oxygenated because the left, it's coming back from the lungs, so it's already been oxygenated, okay? Right, let's do our blood vessels. So we've got this one here, which we'll call E. We'll call this one F, G, and H. So there are 4 blood vessels that carry blood into the heart or out of the heart, and we have to know which one is which. Isabella says the vena cava is E, and that's carrying blood into the right atrium, which will then go into the right ventricle. It's then going to go out and to the lungs. So F is the pulmonary artery. Well done. And we remember that. It's really easy to remember. All arteries go away from the heart, okay? Artery starts with an A, away starts with an A, arteries go away, and it's got to be the pulmonary artery because it's taking blood to the lungs, right? Anytime you see pulmonary, it's relating to the lungs. So G is the pulmonary vein. Again, easy to remember that. Pulmonary because it's coming back from the lungs, and veins, the word vein ends in, and veins always go into the heart. So the blood is going to come in to the left atrium via the pulmonary vein, then into the left ventricle, and then out of the left ventricle. I'll just change my pen colour so you can see it. Out of the left ventricle, so that would be the aorta, which is the main artery, right? So that's carrying oxygenated blood around the body. And I get so many students that can't label the heart. It really is just practice, practice, practice. You should be able to label the 4 chambers and the 4 blood vessels, okay? Might be more simple than this. This kind of looks a bit overlapped with all the colours and everything, doesn't it? But yeah, label your 4 chambers. Don't get your right and left sides mixed up. Label your 4 blood vessels and have an idea of the way in which the blood flows. So it's going in through the vena cava, into the right atrium, into the right ventricle, and then to the lungs in the pulmonary artery. There it gets oxygenated, and then it's coming back in the pulmonary vein, into the left atrium, left ventricle, and then out through the aorta. You don't need to know the names of the valves, but you should know that there are valves, and you should know the function of the valves. So let me circle. You've got valves here, right? These white little flaps are the valves. What is the function of the valves exactly? Stop backflow of blood. And you don't have to know the names of them for GCSE AQA. I don't know whether you do in PE maybe, but you do have valves, and they stop the backflow of blood. They basically stop the blood going back in the wrong direction, so it can only flow in one direction, okay? And the wall, just last thing I should mention to finish, why is the wall of the left ventricle thicker? Why? Why is the wall of the left ventricle thicker? Right, good. You're all telling me the right thing. It needs to contract with more force, right? It needs to contract with more force to create that higher pressure, which a few of you have said, because that's the chamber that is pumping the blood into the aorta. It's got to get all around your body. So the wall of the left ventricle will be thicker. It's got to have a stronger contraction. It's pumping oxygenated blood all around the body. Okay. Good, good, Elizabeth. Well done. Well done, Mary. Okay. Now we've labeled it. The pulmonary artery takes the blood to the lungs to get oxygenated. Remember, it's got to go to the lungs to get oxygenated. Now we've labeled it, let's do a few questions linked to the heart. So for this one, you just have to give the letters on the diagram. This is paper 1 for AQA, Mohammed. That's the difference, you see. So you might want to not revise this if you're at Excel. Which blood vessel transports blood with the highest oxygen concentration into the heart? So we're looking for a vessel that's going into the heart, and the blood has to be oxygenated. So we know it's oxygenated, highest concentration of oxygen. So we know it's coming from the lungs into the heart. So it's got to be D, and that would actually be the pulmonary vein. It's coming from the lungs, so it's oxygenated, into the heart, so it's a vein. It's the pulmonary vein, okay, which is this one. Which blood vessel transports blood at the highest pressure? I think that one's a little bit easier, especially because we've just talked about the left ventricle having a thicker wall, contracting with more force. Yeah, that would be C. That would be the aorta, because the blood is going to leave the left ventricle oxygenated now, because it's the left side of the heart. It's going to go out through the aorta, higher pressure, because the left ventricle has a thicker muscular wall. What is the correct order for blood flowing through the heart to the lungs? So to the lungs, this means it's deoxygenated because it's going to the lungs to get oxygenated. So the first thing I would think when I saw this question is it's got to be the right side. So I can cross off these straight away because it's deoxygenated. It's going to the lungs. It is. It's this one, isn't it? Because it's going to come into the right atrium here, deoxygenated blood in through the vena cava. Then it's going to go into the right ventricle, and then it's going to the lungs in blood vessel B, which is the pulmonary artery. And I know that's right because pulmonary means lungs and artery is away from the heart. Okay. So it's going away. It's got to be option 3. I think I've got a question about the pacemaker actually in a second. Yeah, we're going to do a few microbiology at the end actually. So well done if you got that right. What about the coronary arteries? Well, I can't see them on here, but the coronary arteries supply the heart muscle itself. They're on the outside of the heart. So I can't see them here because this is like an inside view. If I had an external view, I'd be able to see the coronary arteries. They supply the heart muscle itself with blood, which obviously contains oxygen and glucose, so that the heart muscle themselves, the heart muscle cells, can get oxygen and glucose for its own respiration, because the heart needs energy to contract. If it didn't get its own blood supply, it wouldn't have oxygen and glucose for aerobic respiration, and then it wouldn't have energy to contract. Yeah, we've got a big question on stents as well. Oh, it's here. I've actually got a mark scheme to show you for this one because there were so many possible answers that I didn't think I'd be able to write them all down in time. So this is about heart attacks. A heart attack is caused when the heart muscle cells do not get enough oxygen and the cells die. You've got statins and stents, which are 2 treatments used to reduce the risk of heart attack. Evaluate. Ooh, I think this is the first evaluate question we've done. Remember, guys, evaluate means pros, I should say and, pros and cons. Now this is 6 marks. So I would try and do both statins and stents, and I would try and do pros and cons of each. So pros and cons of statins, pros and cons of stents. And in fact, to get 6 marks for this question, you had to talk about both statins and stents, and you had to do pros and cons for both. Does that make sense? So you've actually got an awful lot to think about. Now, remember, a statin is a medication, just basic knowledge. That's a medication that you take every day to reduce the buildup of cholesterol. Well, it lowers your cholesterol, doesn't it? And it reduces the buildup of fat in your arteries. Stents is like a metal mesh tube that you actually insert into an artery to keep the artery open. Put some suggestions into the chat. A-Boy says stents require surgery, statins don't. And you could, A-Boy, extend that. Obviously, if a stent requires surgery, you could talk about the risks of surgery, the risks of internal bleeding, the risks of blood clots, the risks of having an anaesthetic. Statins, though, I mean, I'm immediately thinking you might forget to take them, right? You've got to remember to take them every day. I'm going to show you a mark scheme. Now, I know we're going quite quickly, so I don't want you to write all of this down, but I'm going to suggest, because this was a 6 marker, you would need 3 points for statins and 3 points for stents. So say 2 advantages of statins, 1 disadvantage. Maybe choose 2 advantages and 2 disadvantages to write down just to help with your revision. But for statins, it's just a tablet, right? So it's easy to take. It lowers the blood cholesterol. It means that you don't get a buildup of fatty deposits in the arteries. That means that your heart will keep getting blood. It's a lower cost, but all drugs have side effects. You'd have to take the drug long term. You might forget to take the drug. That's a really easy one to remember, isn't it? For any tablet that you're taking daily, you might forget to take the drug. Then let's have a look at stents. Again, you'd need 3 marks for stents. So 2 pros and a con, or maybe write down 2 pros and 2 cons just for your revision. But with a stent, which is like a metal mesh tube that you actually put into the artery to hold it open, the artery is held open, so obviously blood can continue to flow through. It lasts for a long time. It's quite a common procedure. So it's, you know, I mean, any surgery has risks, right? But it's quite a rapid recovery, and it's quite a low risk procedure because doctors will do a lot of this operation, but there's always risks. So good ones to remember for any operation: risks of infection, risk of surgery. You could have bleeding because you're going into a blood vessel. It could burst. You could also have a blood clot. Yeah, Grace, stents widen the lumen. Yeah, they hold the blood vessel open so it's wider, so more blood can flow through, which obviously is an advantage because if more blood can flow through, the heart can get more oxygen and more glucose for respiration. Yeah, you could give, I mean, they're not all on here, but you could say have to take blood thinning or anti-clotting medication, I'm sure that would definitely be accepted. As a disadvantage of having a stent, you'd probably have to take blood thinning medication or anti-clotting medication. Yeah. ODG, fantastic. I mean, it kind of does, Bianca, because the stent kind of pushes the fatty deposits against the walls. I suppose you could still get more fatty deposits building up, you're right, but the stent, the metal mesh tube, kind of pushes the fatty deposits to the side and helps to widen the artery so more blood can flow through. You don't have to include a conclusion for AQA unless it specifically tells you to, but they don't often do that anymore. That was more old spec. You just do your pros and your cons. This one is particularly hard because you had to do statins and stents, pros and cons of both, if you wanted to get into the 5 or 6 mark bracket. This is an AQA question. Yeah, thrombosis is another word for a blood clot. So for sure you could say risk of blood clot or risk of thrombosis. That would be absolutely fine. Can they rust? I don't think so, because I'm sure, I don't know a huge amount about stents, but I'm guessing they use a particular type of metal that does not rust. Otherwise that would be a big issue. Okay. Statins, well, yeah, statins would take longer to work, so you could have that as a disadvantage of a statin. You'd probably have to be taking it for a few weeks to see the effects of a statin, whereas when you have a stent, immediately that stent is going to be working to widen the blood vessel. Yeah, possibly, Mariam. Yeah. It wasn't on the mark scheme though for some reason. No, Umar, because you're not expected to know that for AQA. Yeah, you can use bullet points. Don't think there's any ethical issues really with this one. Don't think there's any ethical issues. Anyway, these were the main things on the mark scheme. So what I want to see from you guys, I want to make sure you've got at least 6 written down, maybe 8. But when you've got these notes written down, put a plus in the chat because I don't want to spend longer on this slide than I need to. You'd need like 2 pros and 1 con for statins, or 2 pros and 1 con for stents, or you could do 1 pro and 2 cons for statins, 1 pro and 2 cons for stents, but you needed 6, right? And you had to have pros and cons for both. Yeah, they do, Megan. That's a very good question. Examiners do give you credit if it is, you know, biologically correct. They will give you credit. If you know information that's maybe A level beyond the specification, they will give you credit if it is correct. I've even known examiners to literally Google answers to see whether it's actually right, and if it is, they will give you credit. Okay, you could put them in a table. Yeah, that might help you to structure it. There's no harm in doing that for sure. Okay. Right, this is a good question because one of the things that I've noticed AQA do a lot is they link topics, right? And the topics that they link most often are exercise, the heart, and respiration. They go together, and you've got to be able to link your knowledge of exercise, the heart, and respiration. So many people who survive a heart attack get out of breath easily when they exercise. Explain why heart attack survivors get out of breath easily. So if you've had a heart attack, this is quite a hard question. I would say this is going to be a higher tier question. But if you've had a heart attack, your heart contracts less forcefully because basically some of your heart muscle is damaged, right? Some of your heart muscle cells die. So your heart contracts less forcefully, or your heart contractions are weaker. Now, can you see what we're doing here is we're linking together the heart with exercise and respiration. So let's see if you can do that. I wouldn't just say weaker heart, but I'd say weaker contractions would be okay. Bullet points are fine as long as each bullet point is sufficient, right? Let's see if we can now link this. If the heart contracts less forcefully or the contractions are weaker, okay, yeah, we've got some good points there. Let me just add in the one that was maybe missing. Less blood leaves the heart when it contracts. Yeah, think about it. If the contractions are weaker, less blood gets ejected, less blood leaves the heart, specifically from the left ventricle, right? Because the left ventricle is the one that sends blood into the aorta and all around the body. So less blood leaves the heart. Let's continue. What's in the blood? Less glucose and oxygen reaches the cells, because the blood carries oxygen and glucose, right? So if your heart is weaker, if the muscles are not contracting with as much force, less blood leaves the heart. That means your cells are getting less oxygen, less glucose. So why are we getting out of breath easily? Yeah, you could say less aerobic respiration. You could even say, guys, as an alternative, that your cells might have to do more anaerobic respiration, because if they're not getting as much oxygen, they can't do as much aerobic. They may switch to anaerobic. So why are they getting out of breath? You could say, there's kind of two ways to think about this. You could say, well, therefore they're breathing more, they're breathing faster to try and get more oxygen. You could even say to repay the oxygen debt. Couldn't you? They would allow that, because if you're super bright and you've gone as far as thinking, well, if they're doing less aerobic respiration, they're doing more anaerobic respiration. You know that with anaerobic respiration, it produces lactic acid, which needs to be broken down. You might be thinking, oh, they're breathing faster to get oxygen to break down the lactic acid, to repay the oxygen debt. Now, you didn't have to go that deep. That's like super smart students, right? Yeah, this would be higher tier. You don't have to go that deep. You could have just said less aerobic respiration, so they breathe more to try and get more oxygen. But we should appreciate if there's more anaerobic, they also need more oxygen to break down the lactic acid. Okay, we're going to do some more on aerobic and anaerobic respiration at the end. But remember, aerobic uses oxygen and it makes water and carbon dioxide, whereas anaerobic in animals, it no oxygen and it makes lactic acid, and lactic acid has to be broken down, right? And that requires oxygen to break down the lactic acid, which is what we refer to as the oxygen debt. Okay, enzymes are on every year, or there's always something. Why is there weaker muscle contractions? They've had a heart attack. Heart attack literally means some of the muscle cells of the heart have died because they've been starved of oxygen. The heart muscle cells have died, which means your heart muscle is going to be weaker. Okay. Okay. Right. Let's do some more questions then. So scientists have developed patches. This was a follow-up question. Okay. So still about heart attacks in the heart. They've developed patches of beating heart cells to repair damaged heart tissue. So after you've had a heart attack. The patches are placed onto areas of the heart where the cells have died, and new cells grow to replace the dead cells. The patches are made using a person's own cells that are converted into stem cells. Why are stem cells used to make the patches? Now, for this, you need to know what a stem cell is. Stem cells are not specialized cells. It's the opposite, Babatunde. Stem cells are not specialized. They are not specialized. So what type of cell specifically can they differentiate into for this question? Try and make it specific for this question. Yeah, remember we're repairing a damaged heart. Right, so you're right. A lot of you are saying, oh, stem cells can differentiate into any type of cell. Yes, but for this question, why are we using stem cells to repair the heart? Because stem cells can differentiate into heart cells or heart muscle cells, if you want to be. They're called cardiac cells, but heart cells or heart muscle cells. And they will divide or replicate, because it does say here, and I underlined it because I thought it would be important, We need the new cells to replace the dead cells. So the other property of a stem cell, not only can they differentiate into any type of cell, but they will divide, they will replicate or divide by mitosis. So if you put those stem cells, those patches of stem cells onto the heart, the cells will divide to replace the dead cells and differentiate into heart cells. Okay. This one's a really good question, I think. The scientists could have used human embryonic stem cells to make the patches, but can we give two advantages of using stem cells made from the person's own cells rather than using stem cells taken from an early embryo? Socks stuff. You're talking about bone marrow stem cells. Bone marrow stem cells can only differentiate into blood cells. Okay, top answer, I think, in the chat, won't be rejected because they were using the patient's own cells as stem cells, so they won't be rejected. If you use embryonic stem cells from a different embryo, doesn't have the same DNA, could definitely be rejected by the person's immune system. Unethical, we can't just say that, by the way. Guys, we can't just say embryonic stem cells are unethical. What else could we say? How could we explain that? What about something like this? Okay, so remember that, please. If ever you feel like writing, it's unethical, or this is more ethical, can you explain it, please? So in this case, you mean using the person's own cells is more ethical because you don't have to destroy an embryo. Or if you use embryonic stem cells, it's unethical because you do have to destroy an embryo. Or like Mitch has just said, the embryo is a potential life and you've destroyed a potential life. Yeah, cardiac arrest is a heart attack, yeah. Yeah, explain why. And for the embryonic stem cells, the reason it's considered unethical is because you are destroying the embryo, which is a potential life. So explain it, please, yeah. The other one you could say is the adult or the person can give consent, which I saw someone say in the chat before, which was very, very impressive. Genetically identical, yes, Nessa, but the benefit of them being genetically identical is that they won't be rejected. So you'd have to explain that. It's the cells have the person's own DNA, right? So they won't be rejected. They won't be recognized as foreign, so they won't be rejected. That was the top answers on the mark scheme. Okay, that was the top answers. Okay. Less risk of getting a virus. That was not on the mark scheme. I'm trying to think why you're saying that, but I can't think. They could make an embryo that's a clone of you, boy, but the question doesn't say that. So we're assuming that this is a different embryo, someone else's embryo that will not have the same DNA, and therefore the stem cells could be rejected. Both of them need operations though, right? So that's not relevant. Right, let's do some comparisons. This question, I honestly feel like it comes up every year. So I don't know whether tomorrow it might be the first year it doesn't, but I feel like it's literally been on every paper. So I thought I'd better do it. Comparisons of blood vessels, comparing the structure of an artery to a vein, for example. You didn't have to say divide by mitosis, no praise, but they do divide by mitosis, stem cells. You pray it doesn't. No, I wish this would. Like, this is really nice, right? Let's make some comparisons. So arteries have got a narrower lumen. Notice how I'm using comparative language. If I am comparing one vessel to another, I use comparative language. So arteries have a narrower lumen. Veins have a wider lumen. I could do this in a table as well. Arteries have a thicker layer of muscle. Veins have a thinner layer of muscle, or just less muscle in the walls. Arteries have a thicker layer of elastic. Veins have a thinner layer of elastic. We can kind of see where this is going. If you forget to talk about the muscle and the elastic fibers, you can just say for 1 mark, arteries have a thicker wall and veins have a thinner wall. But if you're aiming for maximum marks, you should know that the wall is made of muscle and elastic. Yeah, muscle fibers, elastic fibers, or muscle tissue, elastic tissue. So you can split it into 2 marking points and say that the arteries have a thicker layer of muscle and a thicker layer of elastic. Veins have a thinner layer of muscle and a thinner layer of elastic. Can you mention valves? For sure you can, but make it comparative. So veins have valves. Arteries do not have valves. Don't just say veins have valves full stop, because what's the difference? You have to say arteries do not have valves. Yes, 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 yes. Arteries are thicker, Hannah, because they carry blood at higher pressure, right? So the wall has to be thick to withstand that pressure. Because remember, arteries carry blood away from the heart. It's at a much higher pressure. The wall needs to withstand that pressure. Yeah, that's the reason, isn't it, Bethany? The higher pressure. And the valves, as we said, for the heart, they prevent the backflow. Why is the pressure higher? Because arteries carry blood away from the heart and the left ventricle has contracted with great force. The blood is moving much more quickly at higher pressure in the arteries. Okay. Right, capillaries. Now, there's not much to say about capillaries. Obviously, capillaries are microscopic. These are teeny tiny blood vessels. How would we describe the wall? You don't usually get asked to compare a capillary. You normally get asked to compare an artery with a vein, but who knows? Right, the wall is only 1 cell thick, okay? Why do veins have a wider lumen? Good question, Belle. So there's less resistance to the flow because the blood is at lower pressure. So you need it to be able to flow more easily. So the lumen is wider, so it doesn't like restrict the flow. Yeah, the wall is only 1 cell thick. So substances, e.g., I don't know, oxygen, CO2, can diffuse across the wall. Because remember, it's the capillaries where exchange takes place. So this is where substances are exchanged, right, between the blood and the cells. So oxygen has to go into the cells, carbon dioxide has to leave the cells. They've got to get across the wall of the capillaries. So the wall is literally just 1 cell thick. It's a super thin wall. You've got a short diffusion pathway, all linked together. That's one of the things that I like about biology, is all the topics do kind of link, right? Once you've got your knowledge there. The lumen is very narrow. The lumen is about the same diameter as a red blood cell. So the red blood cells fit through like 1 at a time. It's only about as wide as a red blood cell. And good knowledge there, Isabella. Yeah, the capillaries connect. C for capillaries, C for connect. So the blood goes in the arteries, then the capillaries, then the veins. Lumen is just the hole in the middle. It's literally just the hole where the blood flows in the middle. This is AQA, yes, 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 yes. Laura, can you please tell me if I have a 1 marker but I write 2 things, 1 is right? You'd lose the mark. Yeah, if it said give 1 and you give 2 and 1 is wrong, you would lose the mark. Yeah. So don't. You can give 2 and if they're both right, it's fine. But if 1's wrong, the examiner thinks you're hedging your bets and you're not sure which is the correct answer, so they will deduct a mark. The circulatory system includes the heart, blood vessels, and blood. We're going to do a question on the blood in a second, because I think that might come up. That's not been on for ages. The heart pumps the blood in a double circulatory system. What is meant by double circulatory system? Guys, most students only get 1 mark for this question, and it's worth 2. Can we get 2 marks? Okay, so most students say the blood flows through the heart twice for each circuit. They would only give you 1 for that. What they actually want you to say, so I think this is really important, is the right side of the heart sends blood to the lungs, and the left side, how am I going to finish this sentence? How am I going to finish that? That's how they want 2 marks. Pulmonary and systemic. Yeah, you're right, maybe, Louis. Yes. Around the body. Okay. If you say the blood flows through the heart twice per circuit, they give you 1. But if you actually say, well, you've got 2 circuits, so the right side of the heart sends blood to the lungs and the left side of the heart sends blood around the body, that's what they want for 2 marks. Round the rest of the body, yeah. Okay. Yeah, you could even say the right side sends deoxygenated blood to the lungs and the left side sends oxygenated blood around the body, but you've got to get that clear. If it's 2 marks, okay? Fish do, yeah, you're right, Layla. Good knowledge there. Heart rate is controlled by a group of cells called a pacemaker. Where is this found? Nice, easy 1 marker, but you'd be surprised how many students do not know. The right atrium. Excellent work. So the right side of the heart, it's in the wall of the right atrium. It basically controls the heart rate. Yeah, it controls the beating of the heart. And it's called the pacemaker because it sets the pace of the contractions. Okay. Wall of the right atrium, very specific. Love it, love it. Don't have to do about single systems, actually. Right, I definitely think, well, I say that, I don't know. No one knows. Don't trust people that make predictions, but there's been nothing about the blood components for a while, like a long time. So let's just go through and talk about what is in the blood and what each part does, okay? Obviously use the chat to tell me what you know. Wild, Ashley, this is wild. I'm like getting excited on the stream. Does amylase break down starch? Yes, it breaks down starch into glucose. Right, let's add this. Contain hemoglobin. What does hemoglobin do? Contains hemoglobin, which carries, let's be really specific. I can tell there's more people in the stream because the chat is going wild. I need to watch my handwriting though, don't I? I don't want to get carried away. Okay, so red blood cells, they contain hemoglobin, which carries oxygen around the body. While we're here, just because I think blood could potentially come up, what is an adaptation of a red blood cell? I know that's not on my slide, but what is an adaptation of a red blood cell? Yeah, it's called oxyhemoglobin. Excellent work. When oxygen binds to hemoglobin, you can call that oxyhemoglobin. Good work, Josh. Right, let's just annotate this with a bit more info. No nucleus. Why? So they can carry more hemoglobin and transport more oxygen. Okay. Yes, we are, Nancy, don't worry. And some of you have said a biconcave shape. I'll just draw you it. It's kind of like this, isn't it? Biconcave shape, like a kind of squishy shape. So that's a large surface area, so more oxygen can diffuse in. Because they carry the oxygen, right? The oxygen has to get in. So the bigger the surface area of the red blood cell, the more oxygen can diffuse in, and then it can carry more oxygen, okay? Yeah. Okay, so that's our main adaptations. No nucleus, so can contain more hemoglobin, carry more oxygen. Biconcave shape, larger surface area, again, more oxygen can diffuse in. Hemoglobin is a protein, Kian, that actually carries the oxygen. It's in the red blood cell, and they don't have a nucleus, so they can have loads of hemoglobin. Right, let's move on to white blood cells. Yes, WJP for sure. You should be memorizing photosynthesis and respiration equations. Oh, let's put plasma down here. I missed that off for some reason. White blood cells, lymphocytes and phagocytes. Yes, let's put they form the immune system. That's something basic we should know. They can engulf pathogens. They can make antibodies. I'm actually not going to go too deep on this slide only because I've got another question literally just on white blood cells. Okay, so we'll do a full question on that, I promise you. But the overall gist, just for now, is they destroy pathogens. Guys, just tell me in the chat, what is a pathogen, just super quickly, because the overall kind of general role of a white blood cell is to destroy pathogens, right? To fight infection. What is a pathogen? It's not a disease. That wouldn't get you the mark if you just say it's a disease. I mean, they cause disease, but it's not a disease itself. The definition, some of you are giving me examples, which is good. It's a microorganism which causes disease. Yeah, so your examples would be like bacteria, viruses, fungi, protists, like the malarial protist. Okay. Yeah, but that's your definition if you weren't sure. It's a microorganism which causes disease, and maybe give an example: bacteria, virus, fungus, something like that. Right, platelets. If I only answered with microorganisms. No, you wouldn't get a mark, Ella, because not all microorganisms are pathogens. Like, some microorganisms are beneficial, right? So a pathogen is a microorganism that causes disease. Learn that one. They are fragments of cells. What's their job? What's their job? Yes, they help blood clotting and scab formation. So basically, if you cut yourself, right, the danger is that you're going to get pathogens, you're going to get an infection, right? Because you've basically broken the skin's barrier. Pathogens can get in. So the platelets help to form a clot where you've cut yourself, and then that dries, doesn't it, to form a scab, and that reseals your skin's barrier. So you hopefully won't get any infections in through your blood. You don't need to know about fibrinogen and fibrin for GCSE. That's just like A-level. Wow. Fragments of cells, just ignore that. You don't even need to know. Broken up cells. Plasma. Tell me what the plasma carries. It's the liquid part of the blood. It's actually like a creamy yellow colour, which is mad, isn't it? The plasma is like a creamy colour, but it looks red because there are so many red blood cells in it. Everything else, love that, Parry. It carries everything else apart from oxygen. So it carries CO2, it carries glucose, everything else. I love that. Amino acids, urea, ions, hormones. Yeah, so the red blood cells carry the oxygen. The plasma carries everything else that is transported around your body. Nice, yeah. Okay, perfect. Excellent. I love how you're helping each other in the chat as well. Yeah, antibodies. Yeah, antibodies would be carried in the plasma as well. Urea is not a hormone, no. Urea is produced from the breakdown of excess amino acids in the liver, which is paper 2, so we won't really get into that. Everything except oxygen. Yeah, they carry everything except oxygen. Think of it like that. Yeah, I mean, we've done a load of revision here. This was a 3-mark question, so you actually just needed a function, like carries oxygen, destroys pathogens, and helps blood to clot. But we've just used it to revise the components of the blood, because I was saying at the start of this question that blood has not been on the exam for a while. So I just thought it was worth going through like a description and an explanation for each one. Platelets do help to prevent infection, but it's really important that you explain it's because they form blood clots or scabs, right? So it reseals the skin's barrier. So yeah, it helps prevent infection. Right, communicable and non-communicable. In the chat, can you tell me what these words mean in simple terms? So what is a communicable disease and what is a non-communicable disease? And we've already given me the answer, which is excellent. We're all saying cancer. Cancer is a non-communicable. Yeah, communicable means infectious, so it can spread, right? It can spread from person to person. It's contagious, I suppose. Non-communicable is like cannot spread or not infectious. So you're not going to catch cancer from someone else, okay? Obesity is a risk factor for many non-communicable diseases. Obviously, obesity means being severely overweight. Give another non-communicable disease that obesity is a risk factor for. Now, a risk factor means it increases the chances of getting the disease. It doesn't mean you'll definitely get the disease. Like, I'm not saying if you are obese, you will definitely get the disease, but it's going to increase your chances. Right, lots of you have said diabetes, which would probably be my top answer. Heart disease, some of you said coronary heart disease, which you can abbreviate to CHD. Other ones, you could even say things like depression, that was accepted. You can even say arthritis. Did you know that? Because if you're very, very overweight, it puts a lot of pressure on your joints, particularly like your knee joints and your hip joints, and you can develop arthritis. Coronary heart disease. Yeah. Cholesterol is not a disease, right? Cholesterol is not a disease. You actually have to name a disease. The ones that you would be expected to know for GCSE are definitely diabetes and heart disease, or coronary heart disease, or some of you are saying CVD, which is cardiovascular disease. That's fine as well. Any kind of heart disease. It would be type 2, because type 2 diabetes is linked to lifestyle and obesity, but you could just say diabetes. That was fine. You could say type 2 though, perfect. But you could just say diabetes and get the mark. It says do not refer to the diseases given above, by the way, everybody. So you can't say cancer because cancer was given on the list above. Okay. Right. National policies are used to help people who are losing my mind, who are obese to lose weight. One national policy, so countrywide policy, is that we should reduce the amount of sugar added to our food and drink. Suggest one other national policy. It's got to be a national policy, as in you can't just say people should exercise more. Like, we're talking about things that the government could put in place countrywide. Okay. Add, a lot of you are saying this one, so I'm assuming this is what you've learned. Add a tax on high sugar or high fat foods. So the government could add a tax, right? Like, so the price of a can of full sugar Coke, which they have done, right, increases by like 50p. So if you go to a pub and you order like full sugar Coke, it's going to be 50p more expensive. You could say education, or like you could introduce an education program on health. What else? Yeah, increase awareness or educate people. Like, they could run some kind of education, like in school, like more education on health, diet. The other ones that I think you might be able to remember is like show nutritional info, you know, on food labels. Have you seen that now? Like, every food has got, like, it's in red if it's high sugar or high fat. And you even could have show nutritional information on, you know, like restaurant menus, like takeaway menus, or like if you go into like McDonald's, does it say how many calories now? If you go to a restaurant, Chinese restaurant, does it say like how many calories, how much fat, or how much sugar? Yeah, like in a restaurant or on a takeaway menu. Yes, that's a good one, Cryption. Ban junk food adverts. Ban McDonald's from advertising on the television. Ban, you could even say things like ban supermarkets from putting promotions on high fat foods. Like, they're not allowed to do buy one, get one free on chocolate bars. They're not allowed to do buy one, get one free on full sugar drinks. Do you know what I mean? But these are all things that you could introduce on a countrywide level, right? One burger per person. Stop selling supersized meals in McDonald's. Okay, compulsory PE lessons. I mean, I think that's already a thing, right? I think that's already been introduced. But yeah, this has already been introduced and they allowed that, didn't they? Yeah, so like more compulsory PE lessons for students, something like that. Yeah. Decrease portion size. I don't think that would be accepted because you can't really like control how much portion portion size someone eats in their own home. I don't know. Anyway, there's probably loads of things you could say. It's a little bit of a common sense question, that one, isn't it? Bit common sense. Right, measles is a communicable disease. This means it's infectious, it's contagious, it spreads from person to person. You guys should know that measles is caused by a virus, which is a type of pathogen that, remember, replicates inside your cells and destroys your cells. Describe how measles is transferred from person to person. Guys, this is 2 marks. How are we going to get 2 marks? I've seen some good answers. So coughs, sneezes, even breathing. Even breathe. Like if someone coughs or sneezes next to you, even if someone's just breathing next to you. But what's the second mark? Okay, some of you are talking about droplets. That's what they wanted. Quite hard, no? If someone coughs or sneezes or breathes next to you and they're infected with this virus, then second mark, you inhale the droplets or you inhale the virus. Some of you are talking about surfaces. You could have said touch contaminated surfaces, but you'd still have to then say, and then put your hand in your mouth or nose. Like if you just touch a surface, that's not actually transferring it. But if you touch a surface that has the virus on and then you put your hand in your mouth, then that would transfer the virus. Or you pick your nose or whatever, then that would transfer the virus. So you could have gone for that. But when we think of measles, we think of it as like it's spread via respiratory droplets. That's what we say, respiratory droplets. So you're infected, you're coughing, sneezing, breathing, and then someone else inhales. Yeah? Okay. If your answer is right, but isn't specifically, it depends on the question, Mariam. Some answers, they are looking for very specific things because it's specifically on your spec, right? Other questions, there might be alternatives and you could still get the mark if it is correct. This will be posted after, yeah. Right, who's got a calculator? Guys, we need to make sure we can do percentages. We're going to do a percentage question here. We're also going to do a percentage change question at the end. Dirty needles, no, not for measles. That would be HIV, Zorba. HIV is sexual contact or like sharing dirty needles. So scientists estimate 17% of the UK population have athlete's foot. The UK population is this. Calculate how many people have athlete's foot. All we are doing here is finding 17% of this number. However you do that is up to you. I'm sure we can all find 17%. You could just multiply it by 0.17 to find 17%. Let me check that I typed this number incorrectly. Or you could divide it by 100 to find 1% and then multiply that by 17, which is what I used to do when I was in school, because that was like my fallback method. Is that what we're getting? Yeah, looks like you agree with me. Yeah, so if you struggle with percentages, there's no shame, no shame. You could divide that by 100. I remember getting told in school, if you divide it by 100, you find 1%, right? And then you can multiply that by 17 to find 17%. Athlete's foot fungus grows in moist conditions. Athlete's foot then, remember, it is a fungus. That's the type of pathogen. Suggests one way a person could reduce their chance of catching athlete's foot. Wear flip-flops in public showers. Yeah, you could literally just say dry feet after washing. Or dry your feet after showering, dry your feet after having a bath. Wear socks, I don't think is good enough. You could say things like don't share socks or tights. Like, don't you wear them and then give them someone else to use. Wear flip-flops in public places, or even just like wear flip-flops when you're at the swimming pool. Basically don't go barefoot like in a very public place. Don't go barefoot at the swimming pool. Yeah, this is higher and foundation currently. Use clean socks. Yeah, but remember, we're trying to reduce their chance of catching athlete's foot. We're trying to reduce their chance of catching athlete's foot from someone else. What else? Oh, you could also actually say use a fungicide. I think that was accepted. A fungicide is a chemical that kills the fungus. So you could like regularly put fungicide. I don't think anyone's doing that, but regularly put fungicide on your feet. Got some mad answers in the chat, but these were the ones that are accepted. So the most obvious one, because fungus thrives in moist conditions, right? So after you wash, bath, shower, whatever, dry your feet thoroughly. You could even say use talcum powder. Now that's super old fashioned, but talcum powder is like a powder that really dries your feet. Walk on your hands in the pool. Yeah. Then we might get athlete's foot on our hands, maybe. Cholera is spread usually by drinking contaminated water, by the way. Right. If pathogen, oh, right, okay. This is a good question. If pathogens enter the body, the immune system tries to destroy the pathogens. How does the immune system defend the body against disease? I always bullet point for a 6 marker. Yes. Right. This is actually quite a nice one. This could be foundation or higher. You could get a mark for saying white blood cells. White blood cells. Do you know one of the biggest mistakes that I've seen on this kind of question is people just say your body kills pathogens, your body makes antibodies, your body makes antitoxins. Like, can we be more specific? We're talking about white blood cells, right? When we see immune system, we're talking about white blood cells. Don't just say your immune system kills pathogens either. Don't say your immune system makes antibodies. We're talking white blood cells. Now, if you just wrote this, you're only getting 2 marks. Yes, smart schemes are very specific for biology. That's what makes it quite hard. You're only getting 2 marks. Let's go through. So let's split this now into different types of white blood cell. What do phagocytes do? Good, Miri. Don't say they eat. Please, no. They don't eat the pathogens. They engulf and destroy. They engulf and destroy, or they ingest and destroy would be fine. Yeah, it's called phagocytosis. You don't have to call it phagocytosis though, but phagocytes, which are a type of white blood cell, by the way, if you didn't realize, they engulf and destroy pathogens. Engulf and digest is fine, Mohammed. Lymphocytes make antibodies to destroy pathogens. What else can lymphocytes do? Antitoxins, well done. And you can get a separate mark for saying what an antitoxin does. What does an antitoxin do, everybody? Could you talk about memory cells? I wouldn't really for this question. That would be more appropriate if it was about vaccination and long-term immunity. So we can talk about that maybe at the end. Right, antitoxins, the best word to use is they neutralize toxins. So remember bacteria, and this is important as well, I think, bacteria produce toxins. That's how they make us ill, right? Bacteria produce toxins and toxins damage our cells. Now, lymphocytes can make antitoxins and we say they neutralize the toxins. This is AQA. This question is not about vaccination. It's not about long-term immunity. If it was about like having a vaccination or how do you get long-term immunity, you could mention that memory cells are produced. That would be if you've had a vaccination, right? If you have a vaccination, all of these things still happen. So your phagocytes engulf or destroy, your lymphocytes make antibodies and antitoxins, but memory cells are produced. And then the next time you get the same infection, antibodies are produced much faster and in larger numbers or in higher concentrations. So that would be like what gives you immunity. The fact that you make these memory cells, which kind of recognize the pathogen. They can recognize the same antigen if it's encountered again. And then you can make the antibodies much, much faster and in larger numbers to destroy the pathogen more quickly. Okay, memory cells actually come from the lymphocytes themselves. So you've got different types of lymphocytes, right? But they can actually differentiate or specialize into memory cells. What are antibodies? So remember, antibodies bind to a specific antigen. So if you think about a pathogen is foreign, right? It should not be in our body. A pathogen has antigens on its surface, which are like foreign proteins. Yeah, these proteins are recognized as foreign. They should not be in your body. So that will trigger the immune response. And then the antibody, if I draw one on, try and show you, the antibody is specific. Yeah, the antibody binds to a specific antigen on a specific foreign pathogen. Yeah, you can bullet point, for sure, bullet point. Aseptic technique just means there'll be no contamination, strawberry jam. We're doing a question on that at the end. Okay, so that's it for white blood cells. Could come up for sure. We've got engulf and destroy. We've got make antibodies to destroy the pathogen. They make antitoxins to neutralize the toxins. And just a little reminder there that antibodies are specific. You need the correct antibody to bind to that particular foreign antigen. Right, give one reason why antibodies cannot be used to treat HIV. You could say complementary, yeah, the antibody binds to a complementary antigen. You could say that. White blood cells are phagocytes and lymphocytes. Phagocytes and lymphocytes are white blood cells. Okay. No, we don't do blood groups on AQA. Right, so viruses. Oh, sorry. I gave you a bit of a clue there, didn't I? HIV is a virus. And viruses replicate inside our cells. It's kind of like you're saying the antibiotic, which is a drug that kills bacteria, they can't get to the HIV. The HIV virus is inside a host cell, like it's inside one of your cells. So the antibiotic can't reach the virus unless it destroys your cell. So you need to remember, everybody, the antibiotics kill bacteria only. Antibiotics only, like penicillin from the penicillium mold. Do you remember that one? They only kill bacteria. They don't work on viruses because the viruses live and replicate inside a cell, so the antibiotic cannot reach them. Okay. Chat is buzzing. I can't even keep up with the chat. Would you lose a mark if you said release? No, that would be absolutely fine. Monoclonal antibodies, I have got a question on. Yes. We've done organisation, we've done the heart, we've done digestive system. That's in the organisation topic. Okay, hormones is paper 2 for AQA, so we're not doing that tonight. How long is left? To be honest, however long we need, I'm just going to keep rocking on. I would say we've got at least 40 minutes left because at the end, I do want to do some separate only questions. So, right, guys, this is for you. Can you, in the chat, I know it's a little bit difficult to put an equation, but can you recall, I need a word equation and a balanced chemical equation for photosynthesis, please. I need the word and the chemical. And no, you don't say antibodies neutralize antigens. You say antibodies bind to antigens. And then you say antitoxins neutralize toxins. Right, okay, I can't keep up with the chat, but I can see lots of you. Right, let's do photosynthesis first, right? Now, I think it's important, we have to memorize these equations, right? We just have to. It's like fundamental biology. So we'll do word equation and then we'll do chemical equation. And I'm guessing you've seen these many, many times. You just need to put the effort in to learn them probably. Now, for photosynthesis, we can write light on the arrow and we can write chlorophyll, because remember, we need chlorophyll, which is in the chloroplast, that absorbs the light energy. That's the energy for photosynthesis. Now, in terms of balancing this as a chemical equation, you've just basically got to remember your sixes. Okay. How are we feeling with that, guys? I mean, do I need to explain this, or is it just a case of you guys need to learn it? 666, yes, Nwando. 6 CO2, 6 H2O, and then don't forget your 6 in front of your oxygen. The difficulty is the glucose, right? C6H12O6. Is the arrow writing important? It depends on the question. It's not really important for the equation, but afterwards they might say like, what else is needed? And you would say, well, chlorophyll, because it absorbs light energy. But you don't have to put it in your equation. But then they might give you an equation where there's like a space above and below the arrow, and then obviously they want you to fill that in, right? Chlorophyll is needed. Chlorophyll in the chloroplasts, green pigment, right? It absorbs light energy. That is the energy for photosynthesis. The whole point of photosynthesis for the plant is it makes glucose. It makes sugar, which the plant can then use for many, many different things. I'm definitely gonna go too late here, but can we quickly just whip through the uses of glucose in a plant, please? Because I've just said photosynthesis makes glucose. Glucose is super important for a plant. Do we all know the little acronym? Yes, we do. I've seen SCARF, right. So glucose is used by a plant to make starch. It's used to make cellulose. It's used to make amino acids, so it can make proteins. It's used for respiration, and it's used to make fats and oils. Little side note there, but they could ask, it could even be like a 3 or 4 marker, right? The uses of glucose in a plant. Just remember SCARF. These are all the things that a plant can do with glucose. Guys, if you haven't liked the stream yet, give it a like, because I'm keen to see how many likes we can get. I think we've got like the most people ever on a biology live stream. I think we're breaking records right now. Good on you guys. You're all locked in. Yeah, crops is the same thing, isn't it? Some people use crops, cellulose, respiration, oils, proteins, and starch. It's the same thing. I just use SCARF. Uses of respiration in the body, for sure, Dave. Yeah, because respiration releases energy for muscle contraction, for active transport, for protein synthesis. SCARF is, if you missed that, it's the uses of glucose in a plant. Let's hope now, after doing that little extra, we get a question on that tomorrow. There you go. Uses of glucose in a plant. Can you do metabolic reactions? Well, we're doing it right now, Sam. Photosynthesis and respiration are both examples of metabolic reactions that you could give. A metabolic reaction is just a chemical reaction, right? A chemical reaction takes place in a cell. So these are both examples. Right, can we do now, guys, please, aerobic respiration? Yes, we've got a question on monoclonal antibodies towards the end, but we are going to run late, guys. Obviously, if you want to go, you can, but we are going to run over because I want to try and cover as much as I can for you. Let's do aerobic. Why do they store as starch? Because starch is insoluble. It won't dissolve, so they can store it. Right, so this is the opposite of photosynthesis, basically. So we have glucose plus oxygen, and then we make carbon dioxide and water. And one of the most important things that I want to say to you tonight, why do I put the energy in brackets? Why have I put, this is so important, I can't stress this enough, and I'm really hoping that none of you make the mistake. It's not that it's arbitrary, it's like super, super important. Right, good. Energy is not produced. The examiners are going to hate you if you say anywhere in your exam tomorrow that energy is made, energy is produced, energy is created. Wrong, wrong, wrong. Energy is released, okay? It's not a physical product, as someone just said in the chat. Well done. Energy is released. So whenever you talk about energy in any way, shape or form in biology and any subject, because it's like a fundamental law of science, we say that energy is released. Less energy is released. More energy is released. Energy is released, okay? I've given you the balanced chemical equation. Let's just quickly do anaerobic. There's absolutely loads, isn't there? Now for anaerobic, you don't have to know the chemical equations. You just have to know the word equations. Yes, law of conservation of energy. But you do have to know two, right? Because we've got anaerobic respiration in animals and we've got anaerobic respiration in plants and yeast. So no chemical equation needed, but there are two different equations. Okay, so in animals, it's glucose to lactic acid plus energy, which again, I'm putting in brackets because it's released. And guys, remember, anaerobic releases less energy with anaerobic than aerobic. But it is released more quickly, but there's much less energy released. Let's do plants and yeast. I think I've seen some correct answers for that one. So let's get it down. Glucose, it's different, isn't it? Different products. Ethanol plus carbon dioxide plus energy in brackets because it's released. What is lactic acid? It's literally an acid, right? And it builds up in our muscles when we do anaerobic respiration and it causes muscle fatigue. Muscle fatigue. So remember, if you're higher tier, you should be able to explain that we need to break down the lactic acid. And to break down the lactic acid, we need oxygen, and that's what's called the oxygen debt. The oxygen needed to break down the lactic acid after we've been doing anaerobic respiration. Hassan, that sounds like too much chemistry. Oxygen debt, I can do that super quickly because it's really straightforward. Oxygen debt is the oxygen required to break down the lactic acid. So if our muscles, like say you've been exercising really, really intensely, right? Your muscles do anaerobic respiration because they don't have enough oxygen for aerobic. They produce lactic acid. Lactic acid causes muscle fatigue. We have to break down the lactic acid. And to break down the lactic acid, we need oxygen. And the oxygen debt is the oxygen required to break down the lactic acid. It actually happens in the liver. That's where the lactic acid gets broken down. Okay, so if you're higher tier, that's one for you. Okay, I've literally got that on the screen. The uses of glucose in plants. Have a look over here. We went off on a little tangent. On the right-hand side, just above my head, we've got the uses of lactic acid. Okay. I'm not going to do the eye. Who said that? The eyes paper too. Right, let's do some questions on photosynthesis because we've not done much on this. How can oxygen production be used to show the rate of photosynthesis? I'm guessing you won't know how to answer this question. If we want to know the rate of photosynthesis, what would we have to like measure in terms of oxygen production? This is actually a really hard question. Some of you are saying bubbles, like you could count the bubbles. Can we give a better answer? What about the volume of oxygen? because you'd probably be better to measure it in a gas syringe, no? Because if you count bubbles, there's a lot of human error there. You might miscount a bubble, for example. You're going to miss the bubbles. And you actually don't know the volume of each bubble either. So it's much better to use a gas syringe. You're going to get more accurate data. So if we do the volume of oxygen produced, oh, Vivian, is it Viv? The chat is moving so fast. Vivian, well done. I was going to just ask, how would you get a rate? Now, rate means time, in a given time. So you'd have to do the volume of oxygen produced in a given or in a set time, like 20 centimeters cubed per hour, and then you could work it out per minute. But if you see anything about rate, you have to measure it in a given time. Okay, yeah, because then you do the volume divided by the time to get the rate. Exactly. Right, we've got limiting factors here, guys. Tomato plants are growing in a greenhouse in the UK during winter, obviously, so you can increase the temperature and the light intensity, so they're not limiting factors. The percentage of CO2 in the Earth's atmosphere is 0.04, seems a bit irrelevant, but whatever. Name the factor that is limiting the rate of photosynthesis at point X. Humidity, by the way, pp, is a factor that affects the rate of transpiration, not photosynthesis. Right, it's actually light intensity. Now, there's a cheats way to remember this, or there's a proper way to remember it. I'll give you both. The cheats way, on a limiting factors graph, where you've got like the rate on the y-axis and a factor that affects the rate on the x-axis, the cheats way is where the line is going up, it's whatever is on the x-axis. That is limiting the rate. Wherever it's flat or it's plateaued, it's something else. That's the cheats way. Yeah, so wherever the line is going up, it's whatever's on the bottom of the graph. That's the limiting factor. Where it's gone flat, it's going to be something else, maybe temperature or CO2 concentration. The proper way to remember this is if you learn your definition of a limiting factor. A limiting factor is a factor that, when increased, will increase the rate of a reaction. And if you look at the region marked X, as I increase the light intensity, I'm increasing the rate of reaction. Therefore, light intensity is the limiting factor. Okay? It was literally on last year's exam paper. It said, what is a limiting factor? And the mark scheme said it's a factor that, when increased, will increase the rate of reaction. And where the line is going up, that is true. Because if I'm increasing the light intensity, I'm increasing the rate of photosynthesis, or the rate of oxygen production, which is the rate of photosynthesis. Explain the result. This is quite a hard question now. This is probably one of the hardest questions we've done so far. Higher tier. Explain the results when the light intensity was 0 lux. Have a go, guys, please. Think about this. 0. There's no light. What are we saying? The cheats way, Mariam, yeah, is just wherever the line is going up, it's whatever's on the x-axis at the bottom. That's the limiting factor. Right. Guys, don't just say little photosynthesis. There's literally 0 light. There's no light. There's no photosynthesis. Don't be polite about it. No light means no photosynthesis. Because there's no light. There's no energy for photosynthesis. So no oxygen being produced. Yeah? Because photosynthesis produces oxygen. There's no photosynthesis because there's no light, so there's no oxygen being produced. Right, how is it 4 marks then? Well, have a look at the graph. Not only is there no oxygen being produced, but there's actually a negative rate of oxygen production, which tells us that oxygen is actually being taken in. Yes, there's a negative a negative oxygen change overall. These lines on here, Mariam, where they're going up. Right. So this is how we get 4 marks. Remember, plants respire and photosynthesize. This is where students fall apart. When it's light, they can do both. When there's no light, they are still respiring. Respiration happens 24/7 in all living things. However, they are still respiring, or you could say something like respiration occurs all the time. Something like that. Yeah. And respiration uses oxygen. So there is an uptake of oxygen, something like that. That's why we've got this negative value. Not only are we not producing oxygen, but the rate of oxygen production is negative. It's below 0 because actually they're absorbing oxygen and using oxygen for respiration. Respiration happens all the time. Students just think plants, photosynthesis, and they forget that they are also respiring all the time. When it's bright, bright sun, yes, the rate of photosynthesis exceeds the rate of respiration. But if there's no light, like here, no photosynthesis, still respiration. Do plants also respire? Yes, they do. Yes, they do, for sure. All living things respire. Do you remember Mrs. Gren, like from year 7 biology? Every living thing respires. Like that's what makes something a living thing, is if they can do those 7 Mrs. Gren things. Okay? Yeah, these, Isaac, all these kind of questions that I've done, no, this is combined and triple. All these things that I've done are applicable for combined and separates because like 90% of the content for paper 1 is the same. I don't know if you've looked at your specs, but there's literally only 3 extra things for separate science. Like paper 1 is pretty much all the same. There's like monoclonal antibodies, plant disease and defense, and culturing microorganisms. That's the only topics that are triple only. Okay, right. Again, probably one of the hardest questions that we've done. We've got cardiac output, and they've told us how to calculate cardiac output. Your cardiac output is your heart rate multiplied by your stroke volume. If you don't know what stroke volume is, it's the volume of blood pumped out of the heart with each contraction. It's like the volume that actually gets pumped out every time your heart contracts. And obviously your heart rate is just your beats per minute. Like how many times does it beat per minute? We're not doing meiosis because it's paper 2 and we've already done mitosis, like right at the start. We did 2 questions on mitosis. Calculate the percentage increase in the cardiac output of the athlete after completing the training program when their heart rate increased to 130 beats per minute. Okay, graph skill time. 130 beats per minute. I'm going to identify that on the graph. I'm going to use my ruler, which I can't do on here, but you guys will do. And I'm going to draw a ruled line across so I can accurately read off that value. Okay. So before the training program, make sure I know what line I'm looking at. Before the training program, I can now work out the cardiac output because the heart rate in beats per minute was 130 and the stroke volume was 86. The equation has told me to multiply them together. So I can then get the cardiac output before the training program. Okay. Is that 87? No, it's 86. It's just that my lines on the graph are a little bit dodgy. If you have a look, it's 4 below 90, so it is actually 86. After is nice and easy to read off. So after, I've got 130 beats per minute and my stroke volume is 100. So after the training program, that's what the cardiac output is. Now, I need to do the percentage increase. Okay, so I've only done part of the question. I've only basically worked out the cardiac output before and after. What's the equation for percentage increase, please? Do we need to know max and average heart rate? Not for this question, no. This question, it told us to use 130 beats per minute, so I just needed to read off the stroke volume. Right, it's the change or the difference over the original times 100. Yeah, and you use that same equation, whether it's percentage increase or percentage decrease, it's just the difference in the values over the original value or divided by the original value and then times 100. So let's work out the difference first of all. The difference in these two values. So 13,000 take away 11,180. So the difference is 1,820. Now the original before the training program was this one, wasn't it? So that's my original. That's before they did the training program. So I do the difference or the change over the original, and then I multiply it by 100. And you guys, I've not seen because the chat is moving so quickly, but if you have got an answer, can you put it into the chat for me now so I can see who's getting this right? Well done. I've just seen yours out. Well done. That's a sensible number of decimal places as well, isn't it? Yeah. Yeah. Mine says 16.279. So you could give it 16.28 would be fine. Doesn't say how many decimal places or how many significant figures. So 16.28 would be fine. 16.3 would be fine. That would be fine. Yeah. How did you get 100? Oh, from the graph. So 130 beats per minute. I was going up and I looked at the stroke volume before training and I looked at the stroke volume after training, and that's where I got the 100 from. I worked out the cardiac output before and after using the equation that they gave me. And then I did the difference in the cardiac output over the original cardiac output before training. And there you go. 17.2. Oh, I don't get 17.2, so I don't think you would get the mark for that. Okay. How did you get 86? I read it off the graph here. 81, 82, 83, 84, 85, 86. I read it off the graph. Okay. So many requests for other topics. Let's do this one and then we'll see what requests we've got. An increased cardiac output will provide more oxygen and more glucose to the muscles. Explain how this helps that. Again, guys, this is linking, right? We've got another question that's linking exercise, like muscles and respiration. This is so, so common on paper one. So let's think about it. If they're getting more oxygen and more glucose, right? And don't just say aerobic respiration, more aerobic respiration. Like it's about more, right? If they're getting more oxygen and more glucose, they can do more aerobic respiration. And we're exercising, right? So we're going to say in muscles. So more energy is released. What else are we going to say though? Maybe we could say, so stronger muscle contractions. We still haven't got 4. This is a hard question. What are we going to go on to say? We know they're getting more oxygen and glucose. They're exercising, so it's the muscles that are important. The muscles, if they're doing more aerobic respiration, more energy is released, not produced. This is great because their muscles are going to be able to contract more. Yes. Yes, let's say for mark point 4, less aerobic respiration, so less lactic acid produced, or less oxygen debts because you won't need that oxygen to break down the lactic acid, or won't suffer from muscle fatigue. Yeah, well done, well done. This would be higher, wouldn't it? Because you're going deep. You're going deep, like foundation for sure. You should know that aerobic respiration needs oxygen and glucose. And if they get more of it, they can do more aerobic respiration and release more energy. But we're going a little bit deeper here because it's basically like, if they're getting more oxygen, it's going to delay having to do anaerobic. And that's great because it means they won't produce as much lactic acid, or they won't have the oxygen debt, or they won't suffer from muscle fatigue. Yes, yes, Sinair, excellent, excellent, excellent. More endurance. Maybe if it was 5 marks, yeah, are you? Because if they don't have the lactic acid and the muscle fatigue, then that does mean that their muscles will be able to contract for longer because they won't be suffering with buildup of lactic acid. Nice, 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 nice. I've done oxygen debt already. It's basically the oxygen required to break down the lactic acid that you've made in anaerobic respiration. Okay, right, I've left this blank because I want to just talk to you about an enzymes practical. I don't know, I just have a little feeling. Might be totally wrong. It's not a prediction. We've talked about mitosis, we talked about a microscope practical, which was one of my ideas. We've done a little bit on the osmosis practical, which was another one that comes up a lot. But enzyme practical, I've left the slide blank, so just grab a piece of paper or whatever. I just wanted to talk to you with this example about how I approach planning an investigation, okay? Because you will be asked to plan an investigation. We don't know what on, but I'm going to choose this example. The way I approach it, whenever I'm planning an investigation, and it could be 4 to 6 marks, and if you've been in classes with me before, you will have heard this before. I always think, what is my independent variable and how am I going to change it? Now, let's take an enzyme practical as our example. And let's imagine we're using starch and we're using amylase to digest the starch. What could my independent variable be? What could they ask me to plan? What could they ask me to plan? Temperature or pH, that's what I'm guessing. So my independent variable, if we are asked to plan this, it's probably going to be that we have to change the temperature or we have to change the pH, because they are 2 big factors that affect enzymes. So when I'm planning this, I think, right, what's my independent variable and how am I going to change it? So for example, if I'm changing my temperature, I could say something like use, I don't know, 5 water baths at different temperatures. E.g., I don't know, 10, 20, 30, 40, and 50. 5 is quite a good number to do because it means you'll have enough data to like plot a graph and see if there's an actual trend. Yeah, if it's pH, you would use, I mean, at GCSE, you can just say use pH solutions, but they're actually called buffer solutions if you want to use. So you could say use 5 different pH solutions or 5 different buffer solutions. You could do pH 2, 4, 6, 8, and 10. 5 is a good range for your independent variable. Then I think, and this is how I plan any investigation, what is my dependent variable? This is the one that I'm going to measure. And the reason I've chosen this practical is it's tricky. We've got starch. We're trying to investigate the breakdown of starch by amylase. How am I going to measure the breakdown of starch by amylase? How am I going to measure the rate of breakdown? How am I going to measure the digestion? Right, I'm going to use iodine, guys. I'm going to use iodine. And I'm going to measure the time taken for iodine to stay orange. Why? Can you explain? Do you get your heads around that? I'm going to literally have a little tile and I'm going to put iodine drops, and I'm going to be using iodine for this, right? So I've got a little spotting tile. I'm going to have iodine and I'm going to take a little sample of my solution, right? I don't know, take a sample every like 30 seconds and my iodine is going to be blue-black. Whoops. But I'm going to get to a point, can't see where my pen is because it's too light. I'm going to get to a point where the iodine stays orange. Right, okay, you seem to get that. Basically, listen, this is what's tricky. Amylase digests starch into glucose, right? If I mix my amylase and my starch together, I'm going to take a sample every 20 seconds. Now, to begin with, the iodine is going to be blue-black. Because to begin with, there's still starch present. And iodine goes blue-black in the presence of starch. So I'm going to take a sample after 20 seconds, it still goes blue-black. I take another sample after 40 seconds, it still goes blue-black. There's still starch present. When I get to this point, the iodine stays orange, all starch has been digested. So that is the time that I want to record. Go back to your DV. I'm going to be recording the time taken for the iodine to stay orange-brown because this tells me all starch has been digested. It's all been broken down into glucose. Does that make sense? Yes. Well, Amar, I can't say for other subjects, but for me, the best way I find to structure is I tell the examiner, this is my independent variable. This is how I'm going to change it. This is my dependent variable, and this is how I'm going to measure it. So I'd say I need to measure the time taken for iodine to stay orange or brown because this tells me all the starch has been digested. So you'd take samples, you'd test them with iodine, and you'd record the time taken when the iodine stayed orange because that tells you there is no starch left. The amylase has broken it all down. Then, big important tip for any investigation that you plan, you must give at least 3 controlled variables. Again, using this example, what would your control variables be? These are the ones that you have to keep the same because if you don't keep them the same, they will affect your results. In this case, they would affect the time taken for starch to be digested. Right, so in this one, you could go for volume of iodine. And remember, if you want to get 5 or 6 marks on a plan, you need at least 3. Volume of iodine. Volume of starch. You could even say concentration of starch. This is why most students don't get 6 marks because they forget the control variables, by the way. Volume of amylase. You could say concentration of amylase. And then it depends what you've changed, right? If you change the temperature, you'd have to control the pH. But if you change the pH, you'd have to control the temperature. So I just find structuring it like this is so helpful to make sure that I've included everything the examiner is looking for. Have I done my IV? Have I said how I'll change it? For example, I'll have 5 water baths set up at 5 different temperatures. Have I done my dependent variable? Have I explained how I'm going to measure it? And have I got at least 3 control variables that I have to keep the same so they don't affect my results? Okay. Yeah, listing the control variables is like one of the most important things. It limits you. Like if you've only got 1 control variable, you're like stuck at 3 marks. If you've got 3 control variables, you can get 6 as long as you've got the rest of the method okay. Again, you can use bullet points for a method. It helps because it helps you to step it through logically. You could use numbered steps, but this is like how I start it. And then I might do a few extra sentences just to explain a bit more detail about the method. But the examiner is really looking for what are you changing and how, what are you measuring and how, and what are you controlling your control variables. Yeah, no, I would have talked a little bit more here. You know, my dependent variable, like I would have talked a little bit more so that was clear. For example, mix the starch and amylase together, take a drop every 20 seconds and add to the iodine, and then measure the time taken for the iodine to stay orange. So I would have had to beef it up, right? Just to explain exactly how I'm gonna get to that dependent variable, exactly what my method's gonna be. But this is my kind of outline for how to do an investigation plan. Independent variable is the one you change on purpose to see the effect of that variable. Is this your prediction? My predictions, I might be totally wrong, but enzyme practical, microscope practical, what else did I think? Blood, components of the blood, white blood cells. There's always enzymes, there's always photosynthesis, and there's always respiration linked to the heart or exercise, always. Amar, if you just list the variables, that's not good enough because it's not a method. But what I'm saying is use that as your like crib sheet. So if you think, what's my independent variable and how am I going to change it? What's my dependent variable and explain how I'm going to measure it? Do you see what I'm saying? I'm not just listing the variables, but I'm using those as my cues. I put the variable and then explain how I'm going to do it, right? How I'm going to change it, how I'm going to measure it. Okay. That was just my example, starch and amylase. I just thought it could be an enzyme. Yeah, so you add the amylase to the starch, and then obviously the amylase will start to digest the starch, and then you take like a little pipette sample out, maybe every 20 or 30 seconds, drop it into your iodine. To begin with, the iodine will go blue-black, but you're looking for the time when the iodine stays orange or brown, whatever you call it, because that's the time that tells you all the starch has been digested. Usually 6 marks for a plant like this. No, this is not food tests, but food tests came up last year, and food test is really easy. Literally just learn them. Iodine solution, test for starch, goes blue-black. Biuret solution, test for protein, goes purple. Like, just learn them. Benedict's reagent, test for glucose, heat it, it goes red. Like, I don't even know, like, I can't even explain it. It's literally, you just learn them. You learn the name of the reagent and the color change. The only extra thing is for the Benedict's test, you have to heat it. So you add the Benedict's reagent and then you heat it. And if it contains sugar, it goes red. The others are just straightforward. For starch, you just add the iodine solution. If there's starch present, it goes blue-black. For protein, you add the Biuret reagent. If there's protein, it goes purple. Like, there's not that. Lipids, there's different ways of doing it. You can add Sudan 3 and you get a red layer, or you can add ethanol and water and you get a cloudy emulsion. Okay. A buffer solution, it just controls the pH. So you could use like 5 different buffer solutions to set the pH to 5 different pHs. Guys, we've gone well over time. Don't forget, we have Cram courses for your other subjects. If you've not signed up, if you're not following them on TikTok, get them on TikTok, click the link in their bios, sign up. I think you can even click the link in my bio to sign up to all of them. They are all free. You can go to the My Headspace website as well. If you've enjoyed this lesson, please, please, please leave a review. I have not finished, by the way. This is just the end for combined. We are going to continue because I have got some triple only questions. There are 3 extra topics: monoclonal antibodies, aseptic techniques, and plant disease. We're going to do them. We're going to do a question from each topic. Stay here, stay here. If you are combined, you are free to go. And I'm going to recommend, a lot of you are throwing questions at me like, can we do this? Can we do this? Can we do this? This is not just self-promotion, but honestly, I have got videos and questions on all of these topics on my TikTok. So go to Laura Does GCSE Biology right now. If you want the xylem and phloem, I've literally got a playlist, yeah, with loads of questions on the xylem and phloem. And it will be in playlist 2, I think, organization. Check them out if you want anything. I've got playlists, module 1, module 2, module 3, module 4, which are paper 1 modules. Go in there, scroll through, find the questions on the topic you want, do more revision. Remember, you can watch this back later. You can watch it back tomorrow. Anyway, I need to say good luck. If you're triple, can you stay though, please? Can you just stay? I mean, you don't have to, but I recommend it. Thank you. And please, I'd really appreciate it as well. Obviously, leave me a review, please, on Trustpilot. I'd love to read them. I actually feel like, considering we've done 3.5 hours, this has gone so fast. like, I actually am pretty impressed with your engagement throughout, I've got to say. Please leave me a review. And also, can you please let me know how the paper is tomorrow? Because I'm dying to know. So genuinely, message me on TikTok. Tell me what came up. Tell me if any of the things we went through helped. Let me know. Yes, we are. Yes, 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 yes. No, I would beef it up a bit more. Okay, let's go triple people, combined people. Good, good, good luck. Let me know. Keep in touch. I'll be thinking of you, I swear. All afternoon, I'll be stressing for you. But you can do it. You can do it. You can do it for sure. Leave me a review. Let me know. Can't wait to read them. Bye, guys. Right, should we do some triple only? Let's have a look at this. Bye, combined people. Good luck, good luck. Right, one of the triple only topics is plant disease. Yeah, the video will be open. It'll be there. Don't worry. You can watch it. Thank you so much. Bye. Yeah. Right. Plant disease. Plants infected with aphids may show symptoms of magnesium deficiency. Magnesium deficiency, magnesium ions, right, which they get from the soil, usually by active transport, but that's combined, so we'll leave that now. The symptoms are yellow leaves and stunted growth. Explain how a deficiency of magnesium could cause these symptoms. Right, lots of us are saying yellow leaves is called chlorosis. Can we explain that a little bit more, please? Right, good, Radar. Magnesium is needed to make chlorophyll. So less chlorophyll equals yellow leaves. And this is called chlorosis, which is a term that you should know. So we've covered quite a lot already there, but this is a triple question. It's harder, right? Magnesium, which plants get from the soil, or magnesium ions, they need magnesium ions to make chlorophyll. So if they are deficient, as in they are not absorbing enough magnesium ions, they cannot make as much chlorophyll. Chlorophyll is green, right? It's a green pigment. They can't make as much of the green pigment. The leaves end up being yellow. Marking point 1. This is called chlorosis. Now we need to get 5 marks. So can we extend this? Combined don't need to know this. No, Combined have left the building. Right. Can we explain it a little bit more before we say less photosynthesis? Come on, let's get all our steps. As soon as we see it's a 5 marker, we should be thinking this is really detailed. Less green means more yellow in it. Less chlorophyll means, right, good. Less chlorophyll means less light absorbed. So less photosynthesis. We're getting there. Okay, we're getting there. So 5 marks, remember. Less glucose is produced. Okay, we can still get an alternative mark if less glucose is produced. Why does this lead to stunted growth? Why? What is one of the uses of glucose that you could directly link to stunted growth? Think of the uses of glucose, think of scarf. Which one of those could you directly link to stunted growth? Protein, amino acids, cellulose. Okay. Because they need protein to grow. They need amino acids to make protein. They need cellulose to grow, right? Because cellulose makes up cell walls. If they can't make new cell walls, they can't have cell division. They can't grow. Why not respiration? So you could have said, but it was quite hard to get the mark. They were quite mean. You could have said, if less glucose produced, you could have said less respiration. But then they wanted you to say, so less energy, and they still wanted you to link it. So for example, less respiration, so less energy to make proteins. They still wanted you to link it. Can you see? Because for triple biology, this idea of stunted growth, it's usually about proteins, to be honest. So if you say there's less glucose produced, so less respiration. That wasn't enough. But if you said less respiration, so less energy to make the proteins, they considered that enough. I think that's really, really mean. I don't know why they didn't just accept less respiration, so less energy for growth, but you did have to say less energy to actually synthesize proteins. Because cellulose makes cell walls. So, and obviously when a plant grows, Shanzeh, when a plant grows, it's through cell division. Yeah. If you're making more cells, each cell needs a cell wall. So you would need to make more cellulose to make those cell walls. Okay. No, you didn't have to say cell walls. I would have just said magnesium's needed to make chlorophyll. Because there's less chlorophyll, there's yellow leaves. This is called chlorosis. And then don't forget the light absorbed. A lot of you missed that out. Less chlorophyll means less light absorbed, so less photosynthesis, so less glucose is produced. With less glucose, they make fewer proteins or fewer amino acids or fewer molecules of cellulose, less growth. Scarf is the uses of glucose in a plant. Starch, cellulose, amino acids, respiration, and fats or oils. Oh no, Sarina. Yeah, you need to know magnesium deficiency and nitrate deficiency. So if we just mention nitrate deficiency, remember they need nitrates to make proteins because there's nitrogen in an amino acid, if you didn't realize that. Like amino acids literally contain nitrogen. So they need nitrates to make proteins. So nitrate deficiency equals stunted growth because they can't make the amino acids without nitrates. They can't make proteins. I know. Tell me about it, OG Reality. Okay. Yeah, they need glucose and nitrates to make amino acids. Yeah. Sienna, because amino acids contain carbon, hydrogen, oxygen, and nitrogen. Yeah, that's for nitrate. So that's the other one. Nitrates and magnesium deficiency. Yeah, so for sure, without nitrate ions, they would have stunted growth. Yes, yes, yes. Okay, let's have a look at another question. A farmer thinks that a potato crop is infected with a potato virus called PVY. The farmer obtains a monoclonal antibody test kit. Kind of irrelevant because we're basically being asked how to make monoclonal antibodies. This did not come up last year, guys. So a lot of people are saying it will, but who knows? To make the monoclonal antibodies, a scientist first isolates the viral protein. Describe how the scientist would use this protein to produce monoclonal antibodies. You don't have to know this for combined. Right, let's go through the model answer. Very good start. Inject mouse with the protein. It's called the PVY protein, isn't it? Okay, so this is the protein that you want to make the antibody for. So you're going to inject that protein, which we're told is called PVY, so we can use that abbreviation. We're going to inject that protein into a mouse. Okay, let's be specific. Obviously, when we do this, the mouse is going to have an immune response and they're going to start producing lymphocytes. But when you say collect lymphocytes, I did see 1 year, they didn't give the students a mark for that. They had to say the specific ones. So collect lymphocytes, for example, that are producing the correct or the specific antibody, because you don't just want to collect any old lymphocytes from the mouse, right? Because the mouse has got loads of lymphocytes, but you've just injected the mouse with this specific protein. You want to then go in and collect the lymphocytes that are correctly making this specific antibody, the correct antibody for the PVY. Does that make sense? No, this is separate. It is a B lymphocyte. It is a B lymphocyte, yeah. But you can just say lymphocyte, or B lymphocyte is excellent if you know it. So don't just say collect lymphocytes. You want to tell the examiner that you're going to collect the lymphocytes that are producing the specific antibody, the correct antibody, the antibody for the PVY that binds to the PVY. And then you're going to fuse these lymphocytes, not the antibodies. You'd be surprised. I see so many students saying, fuse the antibodies with a tumor cell. That's not, you're fusing the lymphocytes, tumor cells, to form a hybridoma. And then the hybridoma clones itself many times, or you can even say divides by mitosis many times. Now, this is only a 4 marker, so you can actually stop there, but I always stick on the end, collect and purify the antibodies, because like if it was a 5 marker. Okay, so how do we feel about this? Yeah, collect and purify. Yeah, because in the past that has been a mark as well. So you're injecting the mouse with the protein, the specific protein that they've said in the question. You're collecting lymphocytes that specifically make the antibody for that protein, not just any lymphocytes. You fuse the lymphocytes with a tumor cell because you're going to combine their properties, right? The lymphocytes have the ability to make the correct antibody, and the tumor cell has the ability to divide many times by mitosis. So you fuse them together. So the hybridoma has both of those properties, if that makes sense. It can produce the right antibody and it's going to clone itself many times. So the hybridoma clones itself many times or divides by mitosis many times. You've got all these cells now making the correct antibody. You collect and you purify the antibody. Okay. Yeah, the protein is like the antigen, isn't it? The viral protein is working like the antigen here. So it might be an antigen, it might be a different protein. This is a viral protein. Whatever that protein is, you inject the mouse with it. This is triple higher. This is triple higher. There's very little triple that is foundation. Most of the foundation is the same as combined. But the plant disease question that I just did was foundation. And the one we're about to do now is also foundation. This was a 4 marker. I've only seen it 4 or 5, to be honest. Can we do cancer in terms of this? So the only thing I could say for that is you can attach like a drug or a radioactive substance to a monoclonal antibody. Like if this is a cancer cell, yeah. So a cancer cell will have specific antigens on its surface, like unique antigens, like cancer cells have different antigens, right? So you could produce a monoclonal antibody that could specifically bind to the antigens on a cancer cell. And if you attach some kind of drug or radioactive substance to the monoclonal antibody, it's like you're targeting that cell. The monoclonal antibody will deliver that drug specifically to the cancer cells. So it will destroy cancer cells without destroying healthy body cells. Do you see what I mean? Because the monoclonal antibody will only bind to the antigens on a cancer cell. So the drug or the radioactive substance is going to accumulate around the cancer cells. So the cancer cells will be destroyed, but the normal body cells won't be. Okay. Yeah, pregnancy test is another use. I haven't got a question on that, unfortunately. But hopefully, if it does come up, it's going to be how to make monoclonal antibodies, or maybe now we've talked a little bit about treating cancer with monoclonal antibodies. The benefit of it is it destroys cancer cells without destroying other body cells, whereas traditional like chemotherapy and radiotherapy destroys healthy body cells as well, right? This is targeted therapy just for cancer cells. So you'd make the monoclonal antibodies, Alexander, And once you've produced the correct monoclonal antibody that is specific for the cancer cell antigen, then in the lab, you would attach like a chemical drug or something radioactive, and the radioactivity would destroy the cancer cell. No, humidity is a factor that affects the rate of transpiration. It's not a limiting factor for photosynthesis. All a fluorescent marker would do, iron, is glow. It wouldn't actually destroy the cancer cell. You want to attach something that will destroy the cancer cell, not just mark or label the cancer cell. We haven't done drug testing. No, I mean, I've done a lot of that with my main course, but again, I would recommend if there's something specific like that that we've not had chance to cover, because I appreciate we don't have time to do everything, right? If there's like drug testing that you're stressed about, if you go to my TikTok and you go to module 3, I've got a lot of videos on how to test for a drug, like preclinical, clinical trials. I've got like a big 6 marker on there. So for sure do that after the lesson. There's usually one evaluate question, isn't there? Just remember pros and cons, both sides. Right. I've done pregnancy testing on TikTok as well. If you go into module 3, there's a video on how a pregnancy test works, just so you know. This is the last question we're going to do. A student grew bacteria on a Petri dish. This is Aseptic Techniques. This is culturing microorganisms. This is foundation and higher, triple. Need to remember. The student used aseptic techniques to make sure only one type of bacterium was growing on the agar in the Petri dish. Describe 2 aseptic techniques the student should have used. Decay practical, that's paper 2, no? Right, wash hands is not good enough, by the way. You could say sterilize. In fact, wash, no. Disinfect, words like that, but wash, no. Sterilize is a good word to use. Wash is never really good enough, unless she said like wash hands with antimicrobial soap or wash hands with antibacterial soap or something like that. But you could sterilize or disinfect the work surfaces. You could sterilize the equipment. You could disinfect the work surfaces or your hands. Sterilize the agar jelly. We usually do that by boiling it, by the way. Some of you are giving me examples of how to sterilize the equipment, which is really good. So some of you said flame the neck of the bottle, which is definitely a really good example of sterilizing equipment. Flame the, do any of you know the inoculating loop? Have any of you heard that? You know the little like metal loop that you use to actually transfer the bacteria? That's called an inoculating loop, and you can pass it through a Bunsen burner flame because it heats it up really hot and kills the pathogens. Yeah, so any of those, sterilize the inoculation loop. Yeah, flame the inoculation loop, flame the neck of the bottle. But because this is GCSE, they actually did just accept sterilize the equipment or any example of sterilization. Right, this is a good one. Tape the lid shut, but do not say seal. Why? Why? That's store the Petri dish upside down so condensation doesn't drip. That's correct, but that's not about preventing contamination, is it? That's just so you don't kill the bacteria. That's not about preventing contamination, so that wouldn't get you a mark. Good. Right, you tape it shut, but what you really should do is just put like a piece of tape there, a piece of tape there, you know, something like that. Do not say seal, because the examiners will not give you the mark, because if you seal it, the bacteria will not be able to get oxygen for aerobic respiration and they will die. Or they might have to use anaerobic respiration, which can be very dangerous, okay? You still need to get air or oxygen in there. So please don't say seal. I know that sounds really picky, but it won't be accepted. What about only lift the lid slightly. What about things like that on the Petri dish? What else could we say for that one, guys? Yeah, pass the inoculating loop through a flame. Chelsea, a microscope has a set resolution. You can't change the resolution. You can only change the magnification. Right, at an angle, yeah. So you could say something like, only lift the lid off slightly, or keep the lid off for as short a time period as possible, or lift the lid, but at an angle. Really important, because you don't want bacteria from the air dropping on your agar, because that would contaminate your agar. So you keep the lid over and you just like position it at an angle, or if you do take it off, you take it off really quickly, just for a really short time period. You tape the lid on so no bacteria can get out, because that could be dangerous, and no bacteria can get in, which could contaminate. Sterilize the agar, you could boil it, sterilize all equipment, flame the neck of the bottle, flame the inoculating loop. Yeah, you could work near a Bunsen burner, but you'd have to explain that, I think. As hot air moves upwards. So it's kind of like, it creates this kind of upward current of hot air. So if there's any bacteria in the air, they're going to move up instead of falling on your Petri dish, right? They're going to be carried up, and you're setting up this kind of convection current. This is like physics now. But the hot air rises, carries the pathogens up and away, so they don't fall or contaminate your Petri dish. You flip the Petri dish upside down when you store it, because obviously water is produced when the bacteria respire, and you don't want the water to kind of build up, because it's kind of like drowning the bacteria, right? They're going to drown in the water. They're going to not get enough oxygen. But if you flip it up, the water falls onto the lid, so the bacteria don't get immersed in the water that they're producing in respiration. And then, why is it important? I think I've already said this quite a lot, but it's basically to prevent contamination. I've kind of been saying that throughout my answer to the first part of the question. Yeah, prevent, because you want to grow like one type of bacteria, right? Maybe to test different antibiotics, maybe to test different disinfectants. You want to grow just one type of bacteria. If you get contamination with other bacteria, that's going to affect your results. No, it would have been 1 mark. Hello, A. I think sterilizing surfaces and sterilizing equipment was classed as the same mark for that first question. So you'd need to say tape the lid shut, or only lift the lid slightly, or something different. They did allow sterilize the agar jelly as a separate mark though. Right, bacteria would grow fastest at 35 degrees Celsius, but in the student school laboratory, they were only allowed to incubate the bacteria at a temperature no higher than 25. Explain why. Can we say a little bit more than too dangerous, not safe? Oh, I'm going to give you a perfect answer. Oh, okay, I saw some good ones then, super fast. So 35 degrees, the way the mark scheme words this is promotes the growth of pathogens harmful to humans. Think about it like this, right? 35 degrees is very close to body temperature. Like body temperature is what? 37? If you're keeping the bacteria at 35, that is like the best temperature for human disease-causing pathogens to grow at. And if they spread, it could cause an outbreak of infectious disease. So 35 degrees promotes the growth of, now they were very specific on this, pathogens harmful to humans or harmful to the students. Whereas 25 degrees, you could have said the opposite, and you could have just said it's less likely to grow pathogens that are harmful to humans, that are harmful to students. Don't just say less likely to grow pathogens. You did have to specify this is in a school, we've got lots of people, we've got lots of students. We don't want to promote the growth of pathogens that could be harmful to a human or a student or infectious, right? Incubate just means like keep warm while they divide and grow. Okay. Yeah, we did. We were meant to finish an hour ago. These questions are in the workbook, which is pinned if you want to go back. No idea, Rocky Road. Can't help you there. Yeah, I know, Nwando, but we know triple science is harder, right? It's harder. Stem cells are on this paper, yes, Lauren. So they are unspecialized cells that can differentiate into many types of cell, and they divide or replicate. Okay. I always use bullet points for everything. Rashid, I think I'm going to have to go because I've literally gone like way over time. However, TikTok, Laura does GCSE biology. I promise you there are questions on there about the structure and function of the xylem and the phloem. There's questions about transpiration, translocation. There's questions comparing. So my advice right now, because you need to get some rest, have a half-hour break. Think about any topics that we didn't get a chance to cover that you're still unsure with. And then go and make some notes from those videos. Because I'm at the point now where I'm like, I can't do any more questions. We've done like 1 billion questions. But I promise there are literally videos on everything. So think about what you want to... And also you've got the morning, right? Isn't the paper in the afternoon? Isn't the paper in the afternoon? So you've got the morning to kind of sleep on it and think, right, what did we not cover? What am I weak at? Hopefully this has highlighted things that you're good at and things that you're weak at that you still need to go over. Okay. Please leave a review. Much appreciated, especially from you guys that are triple science. I would love to hear from you. All you need to do is scan the QR code. You can write a review. I will love to read them. Massively appreciate it. Guys, shout out to you for staying pretty much an hour over the planned time of this lesson, but I wanted to keep you for longer. I wanted to do some predicted triple questions as well for you guys. Thank you so much. I've got to say, I was a little bit worried about doing this lesson with so many of you, but it has been amazing. I have enjoyed myself and you have been super focused. So well done, well done. Playlists 1 to 4 on my TikTok are paper 1 for AQA. Topics 1 to 4 is the stuff we've covered tonight, paper 1. Okay. Thank you, thank you, thank you. Like the video, subscribe. I sound like a proper YouTuber. Make sure you join me for Cram paper 2, which is going to be on a Sunday the 8th. In the meantime, check out GCSE videos on TikTok for help. And what else do I need to say? Please message me. Please, please, please let me know what was on the paper and how it goes. Thank you so much. Don't be scared. You have got this. Have confidence, right? Have confidence. They can't ask you questions about things that you are not expected to know. So there'll be something in your head that you can use. There's some stuff on YouTube, but to be honest, Cooper, most of it is on Laura Does GCSE biology on TikTok. I apologize. You might have to see if you can get it just for like a day. Thank you, guys. Enjoy the rest of your evening. I shouldn't say enjoy because you're going to be stressing. Try and get some rest. Try and have a break, okay? You can always come back to it in the morning. It's an afternoon exam, I'm pretty sure. And then I will look forward to hearing from you. A-level biology. Obviously, I recommend it. Yeah, it's challenging, but it's great. It's way more interesting than GCSE. Thanks, Isabel. Oh, lovely. That's what I love to hear. If you do feel like that, Isabel, please write that on a review for me. Makes it all worthwhile for me. Bye, guys. Love to you all and the best of luck. Speak to you all soon. Bye, guys.